So good morning to all. So we welcome you on the second day of our GAN course titled The Exciting Landscape of Precision Medicine and Next Generation Deep Medicine, organized by IIT Indore in association with Purdue University. So I would like to invite Professor Kavita ma'am to start the lecture on the fourth lecture.
Thank you, Leknath. All right, so we start with lecture 4 today. So today I will tell you the difference between sporadic and genetic diseases. So if you check, many times people say cancer is a genetic disease. So that is not a true statement, because only 5 to 10% of cancer is due to problem in genetics. Okay, most of the cancers are sporadic. Sporadic means that there are changes in our body during our lifetime due to the environment, and that can lead to cancer. Okay, so usually you can divide it into two, like genetic, non-genetic. Non-genetic is sporadic, but here they have divided into three categories, and there is slight difference between hereditary and familial cancer. So hereditary means that there is some change in the gene itself, so it goes to the progeny. Okay, because the gene itself has changed, so from parents to children it comes. So that is why yesterday I was saying that in many families breast cancer is very common, right? So most of the women in that family will get breast cancer because they have a defect in their gene and they pass it to their children. Okay, so that is hereditary cancer. Familial cancer is slightly different from hereditary cancer. So familial, this word comes from family. So some family are predisposed to cancer. That could be dependent on where they live. Suppose they live in a place which is really polluted, or they drink too much, all of them, and drinking too much can lead to cancer, right? So I mean, if a person is drinking too much, there is a likelihood of developing liver cancer. Okay, so that's what it says, that certain types of cancer appear to run in a family. These are due to genetic changes and other shared factors. So it's not only genetic, it could be the environmental, because they're living in that place, they have that kind of lifestyle. Okay, and sporadic cancer is completely dependent on environment. So suppose somebody chooses to smoke, so they are basically inviting cancer in their life. Or they are working in a mine, and inside the mine it is severely polluted, right? So that may expose them to chemicals which can lead to cancer. Okay, so this is just to show you that environmental exposures, infection, chemicals, okay, ultraviolet radiation, people who work with radioactivity, if the exposure is too much, normal cells become defective. So this defective cell, if it dies, then it is good, then there is no room for cancer. But if it survives, this defective cell can lead to cancer. Okay, this is a very common one. So this is telling you what is the difference between genetic disease and sporadic. So inherited disease is a genetic disease. You actually are born with a defect in the gene. Okay, and because of this defect in the gene, this person has high likelihood of developing cancer. Okay, sporadic is that we have healthy genes when we start, but eventually we mutate the gene. Suppose you go out in sunlight without sunscreen. Okay, so there are people who sit in the beaches day after day after day, exposing them to sunlight. So that is a sure invitation to cancer, because too much DNA damage because of the UV light. And you know the ozone layer is getting depleted day by day, right? In US, there are some areas which have really bad, like complete ozone depletion. Okay, so there if you go in sun, actually in no time you can feel it gives you a real burnt feeling, right? So that kind of thing is environmental. We have exposed ourselves. Suppose you do not even smoke, but you are in an environment where other people are smoking. So it is very surprising that secondhand smoke is worse than actually smoking. So getting exposed to smoke itself is very bad. Okay, so this is telling you only 5 to 10% are hereditary, 10 to 20% are familial because they are in that environment, and sporadic constitutes the rest of the cancer. This is the same thing shown here. So most cancers, this is good news, most cancers are not inherited. Okay, and if it is sporadic, we can avoid those things. Okay, so that's what it is telling you here basically. So suppose it is a genetic disease, so father carries pathogenic variant. So sometimes you both of the genes, both copies of the genes have to be damaged for cancer to develop. If you have one healthy gene, one wrong gene, the chances are it may not develop, right? So they have children, mother, all the genes are okay. So these children, they have not, when recombination, so these have normal, they have less likelihood of developing cancer. These people, they become carrier. If mother was also a carrier, then some children will have both defective genes and they will be extremely prone to developing a disease, right? So this is the same thing they can show, okay? So this is summarizing the whole thing, that 5 to 10% of cancers are dependent on the genes mutation because it was inherited by the parents. Rest is dependent on environment. Now these are some cancers which are caused by mutations in the genes inherited from parents. Okay, so AML, glioblastoma, ovarian cancer, this is really famous. Ovarian cancer people, okay, they have mutation in a gene called BRCA1 and BRCA2, okay, and BRCA1, BRCA2 are DNA repair enzymes. So if BRCA1 or BRCA2 are missing, DNA cannot be repaired. So why DNA needs repairing? So yesterday I drew the structure of ATP. So you know ATP and guanosine, adenine and guanine, they are purines, right? Purines are attached to the sugar, you guys know that, right? So you will be surprised to know that the purine ring undergoes depurination. So ATP or adenine ring, it just leaves. There's a hydrolysis. So the sugar ring is there. The whole DNA structure is there. So what is DNA made of? Purines and pyrimidines, correct? And it is a phosphodiester linkage which makes the DNA RNA. So in the case of RNA, if it is damaged, we don't care because RNA has a short half-life. 30 minutes, it's gone, right? So even if it is damaged, it's not going to stay for very long. If it makes the wrong protein, protein will also degrade after some time. So it's not a long-term problem. But if the DNA is damaged, it's there forever because DNA is there forever. And the damaged DNA will make the daughter DNA, which is also damaged. So there are many, many, many DNA repair enzymes. They keep on surveying the DNA. They are always moving and checking if there is any problem. If there's any problem, they will fix it. So suppose we go even in this UV light, right? So if you are sitting in the sun for very long, what will happen to bases? If there are two thymine next to each other, the thymines, they will form cross-link. If any two pyrimidines, cytosine or thymine or cytosine, they will cross-link. So DNA will form a bulge. DNA repair enzymes will come, they will fix it immediately. And our DNA is very, 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 very big, right? Because we have so much genetic information. So people used to think, how do repair enzymes know where there is damage? Because something which is so long, right? So actually they have very elegant way. So it's, imagine that if you give a signal at one end like a laser. So if you give a laser here and it comes out at the end, that means the whole thing is intact. But if it doesn't come out at the end, that means something is broken. And that's how the repair enzymes can very rapidly go and fix it. So the point here is that if important DNA repair enzymes are not there or they are defective, that is a sure shot to cancer. Because damaged DNA, if it is not repaired, that can lead to a lot of problems. Okay, so and usually in genetic diseases, many times repair enzymes are defective. So ovarian cancer, like I was saying, see, this is about 19 to 20% of ovarian cancer is genetic, and I just told you only 5 to 10% of all cancers, they are inherited. But ovarian cancer is almost 20%. Breast cancer is towards the higher side, 9 to 10%. Again, due to BRCA1, BRCA2 mutations. And AML, glioblastoma, they have relatively less chance because of genetic problems. Okay. So then why there are genetic changes? The most important is radiation. Okay. So if somebody is exposed to radiation, it will damage the DNA. Smoking is yet another problem. So, overall, lung cancer is number one in terms of maximum number of cases, and that is predominantly due to smoking. Okay, so that is a major problem. Viruses, sometimes you are just a victim there. You are just exposed to viral infection, right? So it's not necessarily it's not that we exposed ourselves. We just got exposed due to some environmental condition. So that also leads to cancer. Okay, chemicals, it is a well-known thing. There are a huge range of chemicals which can induce cancer. So we have to protect ourselves from that. And UV radiation is also extremely damaging, and it can cause cancer. Like I was telling you yesterday, right? This lady had no cancer. She just had a thyroid disease. Doctor suggested you do undergo irradiation, and that she died 6 months later. So basically, she was given cancer in the name of medicine. Okay. So these are some of the cancers. Basically, every single cancer has some component of genetic inheritance. Okay. So these are many different cancers, and it is in the blue they are telling you which genes were changed. So you don't have to worry about which genes. Just to give you an idea that every cancer can be genetic. Okay, small percentage or higher percentage. Okay, but defect in genetics can, these people are more susceptible to cancer. It doesn't mean that they will definitely get cancer, but they are more prone to get cancer. Okay, so BRCA1, so see, normal people who have BRCA1, they have 13% chance of breast cancer. Okay, but if you are lacking BRCA1 greater than 60%, 1 out of 30,000 proteins, 30,000 genes. So we are expected to have 30,000 genes. So one gene is defective or missing, and the chance of breast cancer goes above 60%. MLH1 is also a DNA repair enzyme. So if that is missing or defective, ovarian cancer increases to 20%. This guy, APC, up to 100%. APC is a tumor suppressor. If that is missing, my mistake, MLH1 is a tumor suppressor. No, MLH1 is a DNA repair enzyme. Yeah, this is the tumor suppressor. If this is missing, 100% chance of colorectal cancer. Okay, so I told you about Angelina Jolie yesterday. They both have the same story, Christina Applegate and Angelina Jolie. Did you guys get the PowerPoint, the PPT presentation yesterday? You did not. Okay. When you see Venkatesh, or if I see him, I will remind him to send it to all the students. Okay. And he has them. Okay. And I will send the PDFs of this to him today. And he will definitely send you guys today. So Angelina Jolie and Christina Applegate, so they already knew that in their family they have BRCA1, BRCA2 mutations. So they actually proactively got the surgery done, which they removed their ovary, fallopian tubes, they had breast removal the cancer so the cancer can never come there. Those tissues are missing there. Okay. So so many people, they do genetic testing, especially if their family has history of cancer. Okay. So in genetic testing, they take sample of your cells, they do the testing, and they know exactly. Now it is getting very common. It is very, very common to get this thing done. And then obviously, if someone has cancer, the best way, instead of going for chemotherapy or something, the best thing is to take the biopsy and figure out exactly what's wrong. Why do this person has this disease? And then accordingly treat them. Okay. So you guys have heard of this, what is it called? What is the famous name where you know what is yeah, ancestry.com. Have you heard of ancestry.com? Okay, you haven't heard of it. It is very common in US for the last 10 years or more than that. So people in US, they come from, all over the world, right? So many times people don't know whether they are actually American is just American, right? You are in that country for now, but somebody is from Scotland, somebody is from somewhere, right? So they get their DNA testing and they will tell you that you have 15% Irish blood, and people find some real new thinking. One person got their DNA tested, and they were actually, their ancestors were Native Americans, and she had no idea. Okay, so we have more idea about our thing because I can trace my thing to, so I know I'm from this family and I know all the people there. But there are many people don't know the whole this thing. So this is a lot of fun thing to do. I was thinking if I get it done, they're not going to tell me anything because it's all pure Indian from all the way up to, there's nothing. You will never find anything else in my genetic line, right? But there people find, so this is recreational, right? And it can tell you a lot of things, surprisingly, like if you don't know, right? So there is a politician, I can't remember her name. So she got it tested and she had Native American thing. So when she was going for voting, she was talking to Native Americans that, I have your blood, my ancestors were like that. So that is a different thing. But this is recreational thing, okay? Clinical testing is that actually your doctor prescribes that you have to get your DNA tested, okay? And to make sure that there is no chance of developing a serious disease later in life, okay? So, so one thing is germline testing. Germline testing means we inherit it from our parents. So the zygote, the first zygote has the mutation. So every cell of our body will have that mutation. But if there is some mutation which is at one side, our parents did not give it to us. We actually got it in our life. Okay. So this is just telling you alcohol itself can give you a lot of diseases. I actually know of a person who was very healthy, no problem, but he started drinking alcohol and he died in his 40s just because of alcohol. And some people smoke and some people do both smoking and alcohol. So that's even worse. Okay. So smoking, see how many different kinds of cancer are there. Obesity is also a major cause which can lead to cancer. So cancer is just one thing. Obesity can give rise, obviously, diabetes, right? Please, Venkatesh, you can send these students the PDF files. And tonight I will send you the remaining 4 lectures of today. Okay? Yeah, so obesity can cause many, many, many. First thing we always know is diabetes, right? Diabetes is so easy to get these days, okay? But it also leads to cancer. It also leads to many other diseases, okay? Chemical and environmental factors, these are just, it is, they have studied that many people who are living very close to industrial areas, okay, they are very prone to developing cancer. They are not working there, they are just living close to industrial areas. So they are exposed to all those chemicals, okay. So this is just listing water contamination. In US, I don't remember, I think it was in Michigan or somewhere, water was contaminated, okay, and they even made a movie of that, okay, and it was Oscar winning movie. So people who lived in that area, they were developing cancers, and nobody had an idea because why they were developing cancer, and people were just thinking, so somebody figured it out that they are exposed to something, and the people who were creating all those things, they knew that they are the one who are creating all this waste product which is leading to cancer, but obviously they would not tell, but somebody else figured it out. And so then those, the whole case went on, and they made a very famous movie out of that. So that is what it is.
Madam, I have a question. So, except alcohol and the diet, so 200 years before, so most of the other causes are absent. So
Absolutely.
during that time, the number of cancer incidents were very few and less, or it was not documented. That is why we do not know. Like post-industrial, so all these factors are come.
So there is no doubt about this thing, that our median age has increased, right? So if you see a lot of famous people from 1800 or 1900, many of them died in their 30s, 40s, 50s, right? And even when I was a child, we used to hear in India the median age is in 60s, 63, 64, and then as I grew up it increased to 70s, right? I don't know what is the present one. In US also it is steadily increasing, so people are living a lot longer than before, there is no doubt about it, worldwide, right? It is very easy to go to the 280s these days, right? So this is part of medicine, so many medicines are there, so many preventive medicines are there. So overall, our quality of life is increasing. Initially, if there is even the smallest kind of cancer, no treatment, the person is dying. Now, even if the incidence of cancer are increasing tremendously, there are so many treatments, right? In US, what they do, that they pay you to get your physical examination done, because the companies have realized if you get your physical, so many diseases can be caught before they develop into a disease. So the insurance company, they don't have to pay money for those patients eventually. So they figured it out, pay them to get the test done. And the person who used to work with me, she actually, there were 2 or 3 people who came to me and they told me that they found out during their physical that they already were in the process of developing this disease, and it was caught, it was immediately removed from their body. So to answer your questions, fewer people got cancer those days, but if they got it, they died immediately. And now we can treat them, right? You guys may have heard of your grandparents, or especially females like grandmother, that they had a knee problem or they had leg problem and they had this pain, that pain. Now there are so many preventive ways. It doesn't, you can take things and you will be healthy even in your 60s and 70s, right? So to answer your question, cases have increased, but so have medicines. Okay? All right. So breast cancer can be caused by many different environmental factors. And so this is just telling you. So I want to emphasize something on inflammation. So that is something, normal body should not have inflammation, right? But oftentimes we are exposed to some kind of injury or something which can cause inflammation. So partly inflammation for some time it is okay. If it becomes chronic, definitely it will give rise to a disease. 2 diseases that I work on are definitely caused by inflammation: cancer and Alzheimer's disease. Okay, so inflammation is this stress. Stress is not directly causing cancer, but stress leads to inflammation in the body because cortisol is released, right? And that leads to inflammation. Inflammation will cause cancer. Okay, so and we talked about microbiota yesterday, obesity, alcohol, all of these things, cosmetics, lipsticks that we used. So very common lipsticks, they checked it and they had high chemical contacts which are carcinogenic. Okay, and all the face things that we use may contain carcinogens. So one has to be cautious about that. So this is documented case that thyroid cancer can be caused by pesticides, for examples. So many different contamination, okay, can cause thyroid cancer. So lung cancer, smoking is the biggest thing. Then heavy metal, the miners, I mean, that is their job to go in the mines and clean, but they actually develop many different kinds of disease. Yeah,
So in the case of cosmetics,
yeah.
so is it like better just to avoid it 100%, or there are cosmetics which are natural and not of chemical origin that can be used to protect the skin from UV radiation?
Yeah, so very good question. So anything natural that is good for you, right? So so in India we have a lot of natural remedy, right? Where we don't buy chemicals, right? We make something with haldi and all these things and apply that. So it is better than not applying anything to apply because our skin gets very dry. So if you use high quality oil, olive oil or any good oil, even coconut oil, they are good for you. They have no side effects, and it is advisable to take care of your skin, right? Why? Because skin absorbs directly. So you have to expose your skin to good things. So one should use, but all of these cosmetics means uh, chemical based cosmetics, right? There are so many people who wear makeup all the time. If you are using cream, like good cream, like neem, I'm not going to advertise anything here. It's not meant for advertisement. But if you are using, you know, good quality cream, you don't remove it at night, right? If you use it, it is there. But when people suggest the kind of makeup you are wearing and they said, oh, you have to remove it at night, you obviously cannot. That's not good. That's why they're asking you to remove at night.
Yeah, 25 years ago, even I remember that in South India, at least. So I know that all my sisters or everybody, whoever I see on the road, so they used to apply turmeric paste.
Very
So
good for you.
before when they were taking bath, and that stays for a whole day. But with the time, what happened is almost you see hardly less than 1% of the women apply turmeric paste on the face.
Oh, that is very good for you. If you apply turmeric face thing because it reduces inflammation, it is so good for you. So, yeah, if you're not using it, you should use it. It is that good. But using that is time consuming comparing to taking something commercial and applying on your face. But then you pay the
Because
price.
if you grind it for like few minutes, it is fresh always.
Yeah.
Then you apply.
Yeah.
This is what I have always.
Oh, that is very good for your skin. That's very good. And haldi, the beauty, one thing is that if you consume, right, it goes through the whole thing before it is absorbed by your body. But if you put on your skin, it is directly absorbed. So it's very, very important to do that. Go ahead.
Without boiling, the turmeric will less active component of turmeric will not have the bioavailability.
No, so when you are consuming it, so then it is going inside, it is getting digested and everything before it gets absorbed. If you're applying to your skin directly, right? So, yeah, if it is really, really permeable, it will be extremely good for you, but still, it helps your skin tremendously because you're directly applying it. When you're consuming, it has to get the whole digestion and everything before it is, yeah.
Because it is hydrated, it's not soluble in water very much. When you apply on the skin, the oil that is present dissolves them and takes inside.
Exactly.
This is how it works.
So turmeric is actually very good for you. Turmeric is really, really good for you. And you know, even in commercial things, they advertise it has turmeric in it because it's very good.
Because turmeric.
See, we are advertising. We are advertising, right? The things, see, when I see all of your faces, right, and when I'm telling about cancer, I can see everybody is distressed, right? It is not fun to hear about these kind of things. But you have to understand the truth here so that you can avoid the things you can avoid, and then you know which direction to work to actually, you know, eradicate these diseases someday. Go ahead.
So, obviously, we're speaking about this whole makeup aspect, right? But then now it's advised that you do not put a lot of makeup and you go into the skincare. So, but then the whole skincare industry is also very biased because whatever we use, it has a lot of fragrances and it's highly processed. So, like, what do we really do?
You know, processing is different uh, in different context. Okay, so if you eat food which is highly processed, I can say with very in strong words, yeah, that's very bad for you. So suppose you simply take any vegetable, okay, and you take the vegetable and you make some kind of thing out of it, then you take that and you process it completely, and then you make a roti out of that, and you made the roti and then you process it and you make a powder and you add it in something, like you're heavily processing it. So by the time we eat it, nothing is left there, everything is gone. So sometimes I feel that in just to make something new in food, they are processing it again and again and again and again. Okay, there is one thing, you cook it and you eat it. Okay, long time ago, when actually pressure cooker came, so the advertisement for pressure cooker is that it does not reduce the it is so fast that the good ingredients are still there. You are not killing everything. If you people say raw vegetables, if you eat, they are better for you than cooking it. And if you are frying them and then you are taking the fried one, and forget about the oil content, you have fried them and then you put it in something and then you process it and then you do that, it's useless for you by the time you eat it, right? I was hearing about tomato soup in Italian soup, right? So there is a very famous actor, I can't remember his name. So he also does the food channel. So whenever I went outside and I had minestrone soup, it was so tasty. But when I tried to make it at home, it wasn't as tasty. So he was telling the trick that you have to make the tomato and you have to cook it for at least 1 hour, and the sourness goes away after 1 hour, and then the sweetness comes, and that's why the soup is so good. But then I thought to myself, if I'm cooking tomato for more than 1 hour, there's nothing left of tomato there. And I wouldn't eat it that way. I would rather have the sour taste and at least eat healthy. So processing too much. Now, in context of cosmetics, I don't know how much processing they do, and it may not be bad, because if everything which comes out of that is good, it is okay. Okay, some creams are really good that we buy commercially, and that is a skincare thing. Okay, but if you are using, uh, I don't know much about creams or something, but if you are putting lot of things on your face one after the other, or you are doing that, anything which you have to remove at night or which you are advised to remove at night, that is not good for your skin in the long term. Something you put at night to hydrate your skin, that is obviously good for you, right? So that kind of thing is good for you, but also uh, in skincare routine they add some chemicals which are very bad for you. So, and you cannot trust when they say this is good for you or that is good for you. You have to test it yourself, because whoever is telling, they are just marketing their thing. So marketing is not trustworthy. Yeah, go ahead.
Madam, this radon gas is of natural origin, right? From the earth. This radon gas that is
It could
responsible?
be, I don't know, but radon is also a radioactive material.
Yeah.
So I am guessing that radon could be partly originating from that.
Is it coming from the seabed or from the forest?
I don't know that. I am thinking that these miners, they are exposed to radon gas. But radon is also a radioactive material. So, when I was young, we used to get these watches which used to shine at night because they had radon. So my cousin had it and it used to glow at night. And I was really, really, really excited about that. So I told my mother, I want that watch. And she said, yeah, you will get it. And thankfully, she never got it for me because it causes radioactivity. You're exposed to radioactivity constantly. So, all right. So, and again, if you work with radiation, you have to be very, very cautious about that. Okay. Now, sorry, let me check the time. What time is it now? 10. Okay. I'll go a little bit quickly now. So, this is a root cause for every disease, oxidative stress. Here today we are talking about cancer, but oxidative stress actually degenerates our brain. Okay, so I want to tell you something just to tell you how bad is oxidative stress. So the root cause of oxidative stress is electron transport chain. That's where we use oxygen. And in electron transport chain, electrons are going from one complex to the other. How many of you know electron transport chain? You don't know the electron transport chain, but most of you do, right? You know oxygen is essential for us. And why? Where is oxygen used? It is used in the mitochondria. And this mitochondria is the powerhouse of the cell. Why it is powerhouse of the cell. And why it is powerhouse of the cell? Because it generates ATP. And the reason it generates, the mechanism by which it generates ATP is through the electron transport chain. So mitochondria has an outer membrane. You have seen the picture of mitochondria, right? So this is the outer membrane and this is the inner membrane, correct? So why it has so many invaginations? Because it gives it a lot bigger surface area. And why it needs bigger surface area? Because there are hugely complex proteins located at the inner mitochondrial membrane. So hopefully our mitochondria looks like that. But people who have Alzheimer's disease or Parkinson's disease, mental degenerative diseases, their mitochondria, it looks like this. Like this. So they barely have invaginations here. These are defective mitochondria. So if you have less surface area, then you have less electron transport chain complexes. Now what is the function of this whole complex? They are the one which generate ATP in very simple words, okay? And they generate because electrons are traveling from 1 to 2 to 3 like that. And that generates so much energy, resulting in ATP synthesis. Okay, this is in very, very simple words. So if you do not have enough space to accommodate that, those electrons, they escape. And when they escape, they react with oxygen to form superoxide. Superoxide is damaging, so superoxide dismutase converts it to H2O2. H2O2 is also damaging, so it is converted to H2O. Okay. But if H2O2 is not immediately converted to H2O, H2O2 dissociates to form hydroxyl radical. That will damage DNA, proteins, everything. Okay. So that is very bad. Okay. So it needs to be cleaned up. So when we are born, our defense system is very high. Anytime superoxide is formed, our mitochondria are very healthy. Anytime superoxide is formed, it is cleaned up. So, but as we age, slowly, oxidative stress starts to accumulate, damaging our mitochondria. And because of that, oxidative stress increases. And that is why, as we age, lifetime of accumulation of oxidative stress and damages that we have experienced in our lifetime. So all of that then creates more and more oxidative stress. And that is the root cause. People age. Aging is mainly due to oxidative stress accumulation. The wrinkles that appear on face and body, that is oxidative stress. That's why it says that these creams, they are rich in antioxidants like vitamin E or vitamin C, right? So this is why mitochondria is shown here. So many things, mitochondria can absorb oxidative stress when it is healthy. But if you keep beating on the mitochondria, mitochondria will burst open and then it will create so much oxidative stress. Okay, so oxidative stress can lead to DNA damage. It's the root cause for cancer. It is the root cause for all neurodegenerative diseases. Okay, so this is what it is showing, that I'm telling you just in terms of cancer. So cancer can be caused by many factors, but if all of these factors, they lead to oxidative stress, and oxidative stress leads to initiation of cancer and progression of cancer and metastasis. Okay, so oxidative stress increases mobility. So if the cell is not mobile, it will stay at one place. But if it is very mobile, then it will invade other parts of the body, and that's how cancer spreads in the body. So oxidative stress increases the mode. This is a tumor, but oxidative stress increases the mobility. They break the basement membrane, they go in the bloodstream, they get out somewhere else and form metastasis at a different site. Okay, so this is showing you here. So this is the start of the disease. The cancer cells are formed. Now they are dividing, they are forming a whole tumor. Once they form the tumor, the bad part of cancer is that it never stays in one place. If you don't treat it, it will spread. Okay, so then now some of them are more aggressive than the others. So look at these guys. They are defective cells, but the blue ones are more aggressive, real aggressive. So they then get out of this place, invade the blood vessels, go to some other place and make cancer, a new tumor there. Okay. So, so this is again, so oxidative stress can cause cell death or it can inhibit cell death. Okay, definitely leads to tumorigenesis. It also changes the metabolism. It leads to increased proliferation. Oxidative stress can shut down the immune system. Okay, so immune cells cannot kill cancer, and it definitely leads to aging. Okay, so aging and cancer. So this is, we all know that, right? This is nothing new. Older people are more prone to diseases, including cancer. Okay, so aging and cancer are related, but then there is a term called natural aging and biological aging. So if somebody is 50 years old, that is the age in years. But if the person has a very healthy lifestyle, the body may be a lot younger than 50 years, right? That is called the biological aging. Okay, because if you do the clinical testing for that person may have very healthy body. So that's the biological this thing. So age, so why age is such, so why people who are older are more prone to cancer? Because one of the root causes is oxidative stress that we accumulate. So then oxidative stress, what is the consequence? It damages the DNA. Okay, so DNA repair enzymes will come and repair it, but many times if they're constantly repairing, it will not be repaired in the right way and a mutation will be introduced. And that, so if the mutation is introduced and the cells die, then nothing happens. But if the cells don't die, then they keep on propagating that kind of defective DNA, and that leads to cancer. If it is repaired properly, the cells will remain healthy. So, you know, suppose I will give you my own example. Suppose one airline cancels the flight, right? The flight people can manage all the customers from one airline. But in our case, that day when my flight was cancelled, 200 flights were cancelled at the same time. So in that one room, there were 6,000, 7,000 extremely aggressive, angry customers. And so Air India could not do anything. There were 5 people and 5,000 people, and they were going crazy. But if it was the other way around, 5 employees and 50 people or even 100 people, they would have managed it nicely and everybody would be nice and calm, right? But here they were also fighting, those people were also fighting. This was going back and forth, and people were recording the whole show. Okay, so that same thing for true for the damage also. If the damage is contained, cells can repair it very nicely. But if the damage is constant damage, cells cannot repair it, and then they will make mistakes, and the mistakes accumulate, and it gives rise to a disease. Okay. So what happens with aging? Aging can lead to cancer, and cancer can also lead to aging. So you would ask, how can cancer lead to aging? So suppose somebody has cancer. Cancer will actually change the whole body's homeostasis, okay? And the person will age a lot faster because there is so much damage happening in the body, and the body is trying to fight cancer. So constant stress on the body can lead to aging. Okay, and suppose person has cancer and we are treating that patient, so that person is exposed to chemotherapy. Chemotherapy is very toxic to normal cells. Constant chemotherapy, the person will age. Okay, so that is why cancer itself can lead to aging. All right, so this is aging and cancer are intimately related. But by keeping a very healthy lifestyle, you have met people who are 60 years old, right? I will show you a picture in one of the lectures today, and they don't look 60 years old because they have a very good lifestyle. And there are people who are in their 30s, but they look very, very old because of their own lifestyle. A little bit is genetics, but a lot depends on us. Okay, so this is aging and cancer. They can affect a lot of different mechanisms. They can support each other via many different mechanisms here. Some of them are shown here. All right, so aging causes cancer, and cancer therapy accelerates aging. They are all linked together. And aging limits ability to treat cancer, because if there are diseases and you are giving the drugs, the body cannot take it. A very simple example is that you may have heard of this thing. So when people are diagnosed with cancer, everybody wants to fight cancer, right? And people are very optimistic about it. So they take chemotherapy and they don't feel any real severe side effects even in the first few months, because the body is otherwise healthy, so they can take chemotherapy, right? And then it really helps initially. Okay, sometimes cancer stabilizes, sometimes it also regresses. But if that person is taking chemotherapy for a long time, slowly the normal cells are very tired and they cannot fight it off. And the body's capacity to take the side effects of chemotherapy declines with time. Okay, and then chemotherapy itself becomes so toxic for that person. I have actually heard many people say that, cancer is not going to kill me, chemotherapy will kill me. And then they just stop taking chemotherapy because because they cannot live with that much toxicity. Okay. Okay, so why younger people can fight disease and not older people? So younger people, so there is one other very important factor. You guys have heard of stem cells. We briefly talked about yesterday, right? So we will discuss stem cells in great detail in this course, but there are many, many, many parts of our body which store stem cells, like in the bone marrow and many, many different places. They have their own colonies of stem cells. So we don't have infinite reserve, but we have plenty of reserve. So when our body is exposed to chemotherapy or even some disease, or some kind of damage. Okay, these stem cells, they are very actively coming, repairing the tissue because they have the ability to make any tissue, right? So they are actively going there, repairing it. So our own stem cells can repair the body. But if the damage is constant, then the stem cells are depleted, right? Because we don't have infinite supply of stem cells. So stem cells tend to deplete. And one of the side effects of chemotherapy is that body is so much trying to repair itself from the damage of chemotherapy that stem cells are depleted. And then the immune system also tries to shut it down because they cannot fight it anymore. So this says that when we are young, we can remove dysfunctional cells very quickly. We can maintain fitness. Okay. So because we can remove all the damaged things very quickly, the chances of developing cancer is more or less. But when we are old, we have accumulated too much damage over the years. So we cannot efficiently get rid of dysfunctional cell. And if we cannot, we are exposing ourselves to more diseases. Okay. But when people say age is just a number, in some cases it is true because they keep such a healthy lifestyle that age is in fact just a number because they are not what their actual age tells them to be. Okay, they may be 60 on paper, but they are healthier than a 40-year-old person. Okay, so you can always beat your actual age in years. Okay, so so these stem cells that I was telling you, which have the capability to differentiate into any cells, these are the ones which can save us from all of these things. But if stem cells are depleted, any of these things, all of these things, they can lead to cancer or aging related diseases, right? So that's what it is saying. So it is neurodegenerative diseases. Neurodegenerative diseases includes many diseases. The most famous ones are Alzheimer's and Parkinson, right? So 5% of genetic defects in the genetics can lead to Alzheimer's, but 95% is sporadic. Okay, so there was this real famous Alzheimer's disease scientist. He was a very good friend of mine. His name was Mark Smith. Okay, so I will tell you a little bit about him. So he died at the age of 45, and he was coming from a Christmas party, and some car ran over him, and that person did not stop to see whether he was killed, and then he went home and he killed himself, the driver. But Mark was killed there, and Mark was a very good friend of mine, and so was his wife, Gemma. So what is so special about Mark? He worked on Alzheimer's disease, and he published over 1,200 papers in his lifetime, 45 years of age. And even today, he's publishing more papers than most of the scientists who are alive and are considered very productive because he had so many collaboration. He died in 2010. And every year, 10, 20 papers are coming out with Mark Smith's name on it because his collaboration was so great. And he only worked on Alzheimer's disease, and he was the first person in the world who said oxidative stress is the leading cause for Alzheimer's disease. And you know, people are very, some scientists have very strong ideas about stuff. So when he said in 2000, early 2000, oxidative stress is the leading cause for Alzheimer's disease, the rest of the world used to think Alzheimer's disease is caused by amyloid plaques. He said, no, amyloid plaques is not the reason. Don't waste your time developing therapy against amyloid plaques. So those scientists boycotted him. Okay, they all turned out to be wrong. Okay, so I invited him to come to my place, and he was British, and British people are very fond of Indian food. Like, you know, in London, there are so many Indians. So he was eating Indian curry, and he said he's never going to get Alzheimer's disease because he's eating curcumin here. And he lived only till the age of 45. So the point here is that Alzheimer's is first of all an age-related disease. So people who are in their 70s, they are more prone to Alzheimer's than the people who are younger than 70. 80 suddenly increases a lot. But if you go to 90s, very high chance of developing Alzheimer's. But I know many people who are in their 80s and 90s in India and they don't have Alzheimer's because curcumin is part of our diet every day. Okay, but Alzheimer's, Parkinson, they are definitely age-related disease. Okay, we know Queen Elizabeth lived up to 98. She didn't have Alzheimer's. So it all depends on how you live your life and what kind of diet you are maintaining. Okay, so so that is why aging can lead to many different neurodegenerative diseases. Okay, and so can other disease. So that's just summarizing that cancer risk increases with advanced age. Cancer therapy also induces aging. Okay, and if your body is not healthy enough, cancer is a very aggressive disease. So the treatments are very aggressive, and an aging body or an unhealthy body cannot take that kind of damage that the treatment brings. Okay, I will not go through this thing, but it is basically summarizing the same thing again and again. Aging is leading to cancer, and cancer in turn leads to aging. Okay, so this is just showing that you see the age here. Okay, and most of the cancers increase dramatically as the person ages here. Okay, surprisingly, they have seen, and I don't know the reason, the people who are above 90, who live to be above 90, suddenly their ability to develop cancer actually goes down. If they have survived until 90 years of age and no cancer, then the chances of actually cancer occurring is very low at that age. But we don't know the actual reason for that. That's why it is going down when the person is really, really old. This is yet another way to show. So this is normalized where all different kinds of cancers, the chances of them increases with age. But these are the actual number here. So remember, not all cancers, well, some cancers have many more cases than other cancers, right? So like lung cancer is number one, okay? So this is the actual number of cancers, okay? Actual number of patients who develop cancer per 100,000 people per year. And they have plotted it with respect to age. But the common feature is that, see, cancer is declining a little bit with extremely advanced age. Okay, so here it features some of the things. Okay, I have to stop now, right? Like I'll wrap it up in 2-3 minutes. Okay, so inflammation. Okay, so inflammation increases with age. Okay, so chronic inflammation, this is all the pathways and all the players. Genomic instability, DNA damage, and that leads to genomic instability. The microorganisms in our gut, so intestinal deregulation, and we change our epigenetics with timing. This is just, I will tell you that part later, but this is just cancer survival rate by age. So younger people are more likely to survive, okay? But if this happens, so see 45 years, under 45, more people survive. But as the person is getting older and older, survival also keeps on going down, okay? So this is just the same thing. The majority of so survival generally decreases with age due to comorbidities, means these people have other diseases also. So because of that also survival decreases. Okay. And yeah, this is the picture I wanted to show. So these two people are exactly the same age, but this person exercises. Look at the muscle mass here and look at this one here. And exactly the same age. So that's where the biological age comes into picture. Biologically, this person is a lot younger than this woman, but on paper, they are exactly the same age here. Okay, so this is just to tell you how you can increase your longevity. Okay. So 7, we all know different ways of cutting down cancer. All right. And if it can be detected early, the chances of developing cancer can be really reduced. So prevention is so much better than treating a cancer. And many times for cancer, it is just making the patient's life a little bit easier. Many cancers cannot be cured also, but they all can be prevented. All right, I'll stop here. All right, we can have tea now.
Yeah, okay. We have 10-minute break for the tea, so we'll gather in the same place after 10 minutes.
All right, so today, so the next thing we will discuss is that how genetics can lead to cancer. So this is just to tell you what are these genetic alterations, right? Genetics can cause cancer. So you have a little bit, hey, so I tell everyone, no cell phone, okay? So do not use cell phones while you're in the class, okay? So what happens is that we are constantly exposed, even spontaneously, right? Our DNA gets damaged. So you are sitting in the class, you are not exposed to any bad thing in the class, but spontaneously DNA is getting damaged, and this is just simple chemistry. So you know the phosphodiester linkage, right? So the DNA is linked by phosphodiester linkage, but what happens, just simple chemistry inside the cell, the purine bases are removed. So the whole phosphodiester linkage is there, but purine base is removed. So it looks like, you know, you look at the teeth, right? So all the teeth are attached to the gum, right? And if you remove the tooth, the gum is still there, but the tooth is missing, right? So the DNA is still there, but the base is missing. So these, when purines are attached, they can just spontaneously get hydrolyzed. Okay, the glycosidic bond which is linked to the purine can get hydrolyzed and purine can leave. This process is called depurination. And this happens 5,000 times in every cell per day. So every single cell in our body undergoes depurination at least 5,000 times. The DNA undergoes depurination at least 5,000 times per day in every single cell. So it is immediately repaired. Or we can have some outside exposure. So similarly, there are many processes which can damage the DNA. Similarly, pyrimidines can also spontaneously hydrolyze, but not as much as purines. So there are DNA repair enzymes which can repair it. So you know DNA, so we have two copies of the DNA. So if one is damaged, a single base pair chain in one of the strand, then the other strand serves as a template and it can be easily repaired. Okay, so that doesn't cause bigger harm. But if both the strands are damaged, there are many ways to even fix both the strands there. Okay, so, but many times what happens is that gene itself is amplified. So suppose you are supposed to have only one copy of the gene in the DNA, but there is the DNA itself undergoes amplification. Now you have many copies of that gene in the DNA itself. That is called gene amplification. Sometimes the gene is removed from the chromosome. Okay, that can actually, so P53, we all know P53 is a tumor suppressor. Many times P53 is mutated so that it is no longer functional. Many times P53 is altogether lost in cancer patients. Okay, so gene deletion can cause. Similarly, structure variations. I will show you the examples of all of these. Okay, so I have already shown you this slide. And I have already shown you that if there is a germline mutation in sperm or egg, then the child also inherits it. But if there is some mutation in some part of the body, in some tissue, that is that we have acquired due to the environment, we are not going to give it to the children. Okay? So this is showing you mutation in the germline in this case. So this will get in the embryo, and that embryo, when they develop, they have that disease, and they are also carriers of that disease. But if the mutation is not present in the germ cell and it appears later in life, even at the embryonic stage in some other tissue, they are not going to be the carriers of that disease. Okay? So this is showing you all different kinds of inherited cancer risk. Bone cancer can also be an inherited cancer thing. So basically, every single cancer that is known, it has some genetic component. Some percentage of those kind of cancer can arise from genetic alterations. So now let's look at the mechanisms. What is happening? Okay. So I have made 4 different categories. Oncogenes. You know what are oncogenes? Oncogenes are genes that cause cancer. Okay. So oncogenes. So suppose you so a very famous example is SRC protein, SRC, SRC protein, right? So normal SRC does not cause cancer, but if there is some mutation in SRC and it is always on, always active, so normal SRC will become active for some time and then its activity will go away. But then a mutation can keep it active all the time and that will cause cancer. So SRC at that stage is called a proto-oncogene. So it has that capability of causing cancer. But in normal cells, when it is in the inactive form, it will not cause cancer. But if it is always active, then it will cause cancer. So why? Mutation can make a simple protein which is good, it can make it bad. Okay. Or amplification. Amplification means in the DNA itself you have many copies of that gene. Or translocation. I will tell you the real example of translocation in just a few minutes. Tumor suppressor proteins are often lost or they become defective in cancer. Again, P53 is the best example. Okay. DNA repair enzymes, if they are missing, DNA cannot be repaired. And if DNA cannot be repaired, that is a very big cause which can lead to cancer. And then we'll discuss about epigenetic alterations. So I'm going to focus on these 4 things here. This deals with epigenetics. This is the tumor suppressor. These are the oncogenes and how genomic instability and mutation can lead to cancer. So remember I started the lecture from this slide. These are the 14 features of cancer. Okay. So oncogenes. Okay. So this is a normal gene. I've written it here is an oncogene because what happens is that in normal cells it is active for some time and then it becomes inactive. So it responds. So it is like there is this person. So this person is told you become active, he's doing the thing and he's taking care of that. And when he has to shut down, he's just stopping it. But then this in cancer, this does not respond to outside signal. This is running the show all by itself. So when it is asked, do not divide, it is still dividing. It just is independent of the outside signal. That's when it can lead to cancer. Okay. So suppose there is a protein, suppose there is a cell, right? Let's assume this room is a cell. And let's assume for now that this is the nucleus where I'm standing, okay? And that is the cell surface. And I am the person, I am the oncogene here. So the signal comes, some growth factor comes, the signal is told, it is told to me. Then I tell the nucleus, and the nucleus then responds and the cells divide, right? But I have to get that signal from there before I can tell the nucleus. But then there is something changes in me and I don't have to listen to that growth factor there. So now, whether the growth factor is there or not, I am still active. So I will just give the signal, keep dividing, keep dividing, keep dividing, keep dividing. That's an oncogene because I don't have any shut-off mechanism anymore. I just do not respond to that. Okay. Okay, so this is, and why did I become like this? Because, presumably because of change in me, because I don't have to hear to that. So that is the mutation. So if there is a mutation in the DNA, RNA will also have that kind of mutation, and now this protein will be different because there is a mutation. Do you guys all know what is a mutation? Every one of you? You also know, right? All of you, you also know what is a mutation, right? So that mutation can make me completely active all the time. I don't have to be. I can give you many specific examples. Okay, so I will give you a real example. So this Sarc protein I was telling you. So this Sarc protein has a kinase domain, and ATP binds here and it is active. Okay, so when it is inactive, I'm just making a cartoon. When it is inactive, this active site is closed. I'm just making it like this. But when it is active, this active site is open. Okay, so if the enzyme active site is open, it can catalyze. If it is closed, it will not, this thing. So the mutation can keep it open all the time. Okay, and then it will be active all the time. Okay, so but in normal cases, this is closed. Why it is closed? Because there is some conformational change here which keeps it closed. When the growth factor signaling comes, this protein changes conformation, it opens up. But if you make a mutation and always keep it open, it doesn't need any outside signal. So that is what it is saying, mutation here. See, under normal circumstances, you should have one copy of the gene in the DNA, but now it is amplified, so you will always make more protein. So once again, I want to tell you, if many times our RNA is defective, because RNA is made from DNA, RNA polymerase makes the DNA, and many times it puts the wrong base because it's doing it fast. But if RNA is defective, no problem, because RNA has a short half-life, it will get degraded. If RNA is defective, it will make the wrong protein, and then protein itself will get degraded. Half of the time, if the protein is wrong, protein doesn't fold properly, and if it doesn't fold properly, the cells will immediately clean the misfolded protein. So it will not even damage us at all. Okay, but if DNA is damaged, and if DNA is mutated, every single time the same RNA will be made. And even if that RNA dies, some other RNA will have the same problem. So we can never get rid of that problem. That is why DNA damage is bad, RNA damage is not bad at all, right? So I can change my statement. I always say never in biology, in science. In science, you never say never because there are always exceptions to every rule. So I take back the sentence that RNA damage can never be bad. RNA damage can also be bad, but DNA damage is worse. Okay? And then chromosomal rearrangement. Okay? So I will give you the example of this thing. Okay? So chromosomal rearrangement. Do you guys know what is a promoter of a gene? You don't know that promoter of a gene. That's okay. I just want to. So, do you know, Leknath, what is a promoter? So, let me give you a quick crash course in biology here. So, DNA makes RNA, RNA makes protein, right? All of you know that part. Okay. So, this is DNA. DNA makes RNA, and RNA makes protein. Okay. So I would earlier, not right now. So we have 3 billion base pairs in humans, and people used to think that only 5% of that makes proteins. So we have 3 billion base pairs. So let's say that is our 100% DNA. So this is true that 5% of that 100% DNA makes protein. So what is happening with the 95%, right? So people used to call it, scientists used to call it junk DNA. If we cannot figure out something and we think we are very smart, we call it junk. So the scientists, and you go in the journals, they call it junk DNA. That 95% of human DNA is junk DNA, 5% is this. So whoever labeled this junk DNA really thought that nature was not as smart as that person because nature has made 95% junk DNA. Now we know that is not true. This is something which has come in the last 10-15 years. Okay. So, because I have myself taught in 2004 that 95% of human DNA is junk, because that's what we all believed in 2004-5. So that 95% also makes RNA, but what is still true is that they make RNA, and these RNAs have different roles, but these 95% are still not making proteins. So they are not also protein encoding DNA, but they're making RNA. You guys have heard of microRNA and all these things. So that they are coming from those. And they are called non-coding RNA because they don't make proteins. Okay. So then the 5% which makes protein, or all the RNA that is made, it's not 100% DNA. We can say safely that at least we don't know as yet that 100% DNA will make 100% RNA. Only small percentage will make. So how do cells know which RNA they should make and which RNAs they should not make, right? So DNA has this region called promoter. Okay. So when RNA is made, the enzyme which makes RNA is called RNA polymerase. RNA polymerase. So this RNA polymerase will recognize special sequences on the DNA which are called promoter. It will bind here and then it will make the RNA. That makes sense. So that means promoters are very important because they are the one which will tell this RNA should be made. Okay. So if the promoter region is hidden, RNA polymerase cannot access it and that RNA will not be made. So the thing which is written here, enhancer. So these are the proteins which will expose the promoter region so that RNA polymerase can make RNA. So if it is super exposed in cancer, then you will make a lot more of this RNA. And if you make a lot more of this RNA, you will make a lot more of this protein, and that can be bad. So that is what happens in diseases. Something which is not supposed to be that exposed can get exposed, or for tumor suppressors, it will get hidden. So it is RNA is not made. Is it making sense? So that can also lead to abnormal protein expression. Okay. So here, this proto-oncogene means this gene has the ability to act as an oncogene if it is overexpressed. Okay. So there is some mutation, now the protein is abnormal. Or see, this is what I was telling you. This, let's say it is the promoter region. There is something in there. So now you make a lot more protein. You are not supposed to make it, but something happened here, so it is more exposed, more protein. Okay. So here there is, okay, here we have added either a new promoter, which is very strong promoter. So now, so you know, not all promoters are equal. Okay, some are strong, some are weak. So suppose you open up a cell. Have you guys heard of proteomics, 2D gels? You have heard of it. So I used to show a picture of a 2D gel. If you have ever seen a picture of a 2D gel on the web, have you seen that? Have you seen any 2D gel picture? Anyone? Okay, so suppose every protein appears as a dot, right? The beauty is this, that if it was equal, if all the proteins were equal, every dot will be equal in size, right? But every dot is unique. Some is big, some is small, right? So you guys agree with me, not all proteins are expressed at equal levels in the cell. Some are present in very small amounts, some are present in a large amount, right? And it has to be that way because some proteins are so powerful that they're expressed in very small amount for a very short period of time, and they do their job and they're immediately degraded. And these proteins, if they stick around for a long time, that's a very bad thing. Okay. So what happens is that here something happens and this makes it very strong promoter. So a lot more protein is produced. That's bad. That can lead to, because this is a proto-oncogene, this has the tendency to become cancerous if it is present in huge amount. And see, all of these are leading to huge protein production. So now it becomes an oncogene. Some people have inherent quality to become bad, right? If you keep them in check, they are okay. But if you give them too much power, that is bad. That's the proto-oncogene. Okay, so see, gene amplification, huge amount of protein, cancer. Okay, so that was oncogene. Now, tumor suppressor genes are the good guys. They keep tumor in check. So here, what you want to do, we don't want to do, but cancer, if they are lost, cancer develops. Okay, so here there are many tumor suppressor genes. So when these tumor suppressor genes are there, they keep the cells from dividing too much, and because of that, cancer never develops. But if they disappear, then if they are defective, they are there, but they're not able to do their job, cancer will develop. Okay, or uh, this is let's stick with we'll not stick go to the oncogene, we'll just look at the tumor suppressor. So either they get deleted or they are defective, and that is a root cause for cancer. Okay, so here a tumor suppressor gene is there, and some kind of outside environment, like even secondhand smoke. Okay, secondhand smoke or radiation or UV, this thing. Now the tumor suppressor gene, TSG stands for tumor suppressor genes. So because of that, there is a mutation. They are not functioning, and cancer, it becomes cancer. P53 is, as I said, the most famous example. So P53 is lost or defective in many, many, many different kinds of cancers. Okay. So then let's come to DNA repair gel. So our DNA is getting damaged all the time, and it is getting repaired also all the time. Unless we expose ourselves to really severe conditions where DNA is damaged. So right now we are here, and in our cells deep urination is occurring. That's a normal thing. It can be repaired, but if we go in the beach and we sit there without any sunscreen for hours and hours, the damage can become so extensive that the body's repair system is so overwhelmed that it cannot repair it completely. Okay, and that can lead to disease. Okay, so accumulated DNA damage. If it dies, good news, right? But if it doesn't die, then the cells become very aggressive. If stress is there, and if they die, that is good. But if they just keep on taking the stress and they modify themselves, then that is when a disease develops. Okay. So this is here. So it shows you type of DNA damage which is occurring in our body under normal circumstances, and this is all normal processes which is occurring. So we can deep urination. The purine is hydrolyzed. It can be simple deep pyrimidation, also pyrimidine is also hydrolyzed. Cytosine, you guys know, has an NH2 group. NH2 group can get hydrolyzed to oxo group, keto group, so deamination. Single-stranded breaks, not big deal, because the second strand will act as a template, it will be fixed. Base oxidation, guanine has a hydroxyl group, it can be fixed. This is happening all the time. Every time we are going out in the sun for a long time, even taking long walk in the sun, thymine will form cross linkage like this, or cytosine can form cross linkage, or cytosine and thymine can form cross linkage. But then this light, this simple visible light, not sunlight, simple visible light will reverse that. It will be, that is the easiest fix. So this is not damaging at all. And then sometimes DNA repair enzymes can put the wrong base. It can be fixed, but if it is not fixed, it can lead to mutation. Okay, base alkylation. So these are many different kinds of defects which are being repaired in our body. But if you're constantly exposed to this thing, that's when body gives up, and the body cannot repair this thing. And if it is not repaired, it can give rise to cancer. Okay. So, and diet comes everywhere. Okay, so these kind of problems can give rise to DNA damage, and although the body repairs it, our exercise and diet, these two are big things which can prevent DNA damage from DNA damage. Okay, so this is the antioxidant defense system, DNA repair system. And usually body has this safe mechanism that if it cannot repair it, the cells will die. It will turn on the warning signal for cell death and the cell will die. But then if the cells resist death, those are the cells which are the dangerous cells. Okay. Now let's talk about BRCA1. You have heard of BRCA1 since yesterday. So what exactly happens in BRCA1? So BRCA1 and BRCA2, they are DNA repair enzymes. And they are so famous because they are often deleted in cancer. Okay, so if DNA is damaged, BRCA1 can repair it, but if BRCA1 is absent, DNA damage goes unrepaired. And so this BRCA1, BRCA2, if they are defective or they are absent, it can lead to many different diseases. The most famous one are breast cancer and ovarian cancer. Okay, so that was about the DNA repair enzymes. The last thing is the epigenetic reprogramming. So do you guys understand epigenetics? I told about it a little bit yesterday. I'm going to tell you about a lot more today in the lectures. But epigenetics is that we do not change the DNA. So I will tell you something. Some of you guys know about Alzheimer's disease, right? So in Alzheimer's disease, patients lose their memory, right? More than the age-related memory. So when people get older, some people, not all, some people, they have age-related memory loss. So they often say, oh, I cannot remember this or I cannot remember that. So that is not a big deal. That is just age-related loss. But few people lose memory at a much rapid rate, okay? And that is most likely due to Alzheimer's disease. But even those Alzheimer's patients, if you know of any such cases, Alzheimer's patient will remember their children. Okay, they will remember their children. So Ronald Reagan, obviously you know Ronald Reagan, all of you. So he had Alzheimer's disease. Up until his last day, he did not forget his wife. He was so happy all the time to see his wife. He never not recognize his wife, but all the other people, he forgot that he was the president of United States. He forgot all his colleagues. He forgot, but he did not forget his wife and his children. So these people have long-term memories. All the things they did in their childhood, they will tell you all the childhood things. Okay, they will tell you all about their old thing, but they would not be able to tell that, you know, whatever story you shared with them yesterday or today, short-term memory is lost. So people were very curious why, even in many cases, why people can recall long-term memories and why they cannot recall something which happened yesterday, day before yesterday, or even a year ago. So then I was in a conference and they presented a paper many, many years ago. It is due to epigenetic modifications. So the long-term memories, the DNA has epigenetic changes and they make the long-term memories. And those epigenetic changes do not occur in short-term memory. And we cannot recall those things many times. Like many of us, I mean, you guys are very young, but the childhood stories we still remember. But what we did yesterday or the trivial things of yesterday we have no recollection of those things. So that is also due to epigenetics. So it is very important. So the gene is there, but epigenetics decides whether the gene is going to be expressed to make proteins or not. So that is why epigenetic modification. So it's not surprising. Cancer hijacks the epigenetic machinery to express the proteins it wants to be expressed or to suppress the expression of good proteins and increase the expression of bad proteins. Okay, so now let's talk about what exactly is epigenetics. Okay, so so if we take all the DNA from our body and from one chromosome we take the DNA, and chromosome is what? Highly condensed DNA along with proteins. So chromosome is made of two, we know our DNA is stored in the form of chromosomes. So chromosomes is made of two components, DNA plus proteins. And this DNA, so we have problem in really cleaning our house, right? So many times we clean it properly, many times we even dumps things when we are in a hurry, right? Cells cannot dump DNA. If you take a chromosome and you unravel it, so it's very nicely packed. If you unravel it, one single chromosome DNA can extend to 2 meters, and we have 23 chromosomes. So that much amount of DNA is present in the form of chromosome, and it is packed inside the nucleus of a cell that we cannot see with our eyes. So in a very, very small area, that much DNA is there. But the cells do not have a choice of aggregating all the DNA and just making it a compact ball and just putting it in the nucleus. It is highly organized. Why? Because our cells are dividing all the time. So the DNA needs to be replicated 100% almost every 2-3 days. 100%. So the 100% of DNA has to open up, get replicated. That's one thing. But the same DNA is making RNA, right? And RNA is being made every second in our body. So, and it has to decide which RNAs to make. So DNA is opening up, closing, opening up, closing. So it's an extremely organized structure. You cannot just aggregate the DNA and just make it small just to fit in the nucleus. It's nothing like that. It's a highly organized structure. So this is what it is showing you here. This is chromosome. You open, this is here, it appears that this is just this. No, there are huge level of organization here, huge level of organization. I'll show you different pictures. But when you open it, after a lot of organization, you will come to this kind of structure. And each of these are histone octamers. So his DNA is negatively charged because of the phosphodiester linkage. And these proteins have lysine and arginine, so they are positively charged. So this is an octamer of histone protein, and DNA wraps around it. So it's like you are wearing something with beads on it, okay? So it looks like beads there. And then you open up all the DNA, you unravel it, and you will see the DNA like this, okay? So this is highly organized structure. So this is chromatin here. So some part of chromatin are more open, so those DNAs are more open, some are highly compact. When DNA gets replicated, no matter what, DNA is always replicated 100%. It cannot be 99%. If it was 99%, we would not live to the age we live up to. DNA has to be replicated 100%. Okay, but some part are more open than the other. So those parts which are more open, those are the part which makes more RNA. The parts which are closed, they don't make those many RNAs. They are replicated, they are still replicated, but they don't make as many RNAs. And that is, so what decides that some part of the DNA should be closed, some part should be more open, and that's where epigenetics comes into play. So DNAs, they undergo modification, and the most common modification is methylation of DNA. So this is cytosine, and cytosine can get methylated at this position here. I will tell you the consequence of methylation. So once the DNA is synthesized, you know, when so when you have cytosine, guanine, cytosine, guanine, cytosine, guanine, cytosine, guanine. So if you have too many CG, those C's, not the G's, they have a tendency to get undergo methylation. So when they are methylated, so then they form 7-methylcytosine. So you know the sequence we read. If cytosine is followed by G, we call it CG. But long time ago, maybe 50, 60 years ago, people used to call it not CG, but CPG. Why they call CPG? P is the phosphodiester linkage, which is telling you that they're linked by this thing. You know, like, you know, when I was growing up, we started abbreviating a lot of things, right? Now you guys are abbreviating even the abbreviated version, right? So, so I have to say, oh, that's what you mean. Right now, nobody says billion. It's too hard to say billion, so they call it bill. The next generation will not even call bill. It will be more abbreviated. We cannot even think what they are going to call. So you know it's getting abbreviation, our abbreviation itself are getting abbreviated, right? So those time people were calling CPG, now we call it CG because that's understood. Of course, it is a phosphodiester linkage. But if you look at the textbook, it will say CPG, CPG, CPG. Those are CG, CG, CG. And they are the one which are getting methylated. So I'm just telling you the most common modification. So there is methylation, there is acetylation, and deacetylation. So remember, there are proteins, histone proteins, and DNA is wrapped around it. So the most common changes in DNA is methylation. So arginine and lysine residues in DNA, they can get methylated. For histones, they can get methylated also, they can get acetylated also. Acetyl group is added. And then the acetyl group is also removed here. So this is lysine side chain, and this lysine is acetylated. But lysine can also be methylated. Okay, so look at the colors here. So I have made acetylation green because acetylation opens up the DNA, and DNA is more accessible. Methylation closes the DNA. Okay, so acetylation, it opens, acetylation occurs not in the DNA. Acetylation occurs in the histones, and DNA will be more exposed. Okay, I will tell you more in details in just a minute. Look at the signal here. So here, this acetylation is not occurring in the DNA, it is occurring on the histone protein. And look at this, this is a green signal. So see, I just told you about the promoter region. So when it is acetylated, the structure is more open, and promoter region is this one, it is C exposed here. So now RNA polymerase can bind here, and the RNA will be made. But here it is acetylated. So this is the normal form. When it is acylated, it opens up. Okay, but when it is methylated, see how close it is. It's even more compact, because once it is methylated, these proteins come and bind and completely close the DNA. Now RNA polymerase cannot reach there, and these DNAs will not make RNA. This part of the DNA will not make RNA. So methylation is red signal, acetylation is green signal. Okay, so look at this thing. So it is showing you two parts, protein part, DNA part. When DNA is methylated, it is closed DNA. That does not make RNA. Okay, when it is not methylated, it makes RNA. Histone, when it is less acetylated, less accessible. When it is acetylated, more accessible. Okay, so because of that, so now you can guess yourself. If I have to ask you, I'm supposed to ask you questions during tutorial, but I'll ask you now. So suppose cancer has to play with epigenetics. What will it do to the oncogenes in terms of epigenetics? Tell me that. Anyone? Of what genes? Acetylation of which kind of genes? Oncogenes. Very good. It will do acetylation of oncogenes because then it will be more expressed, more RNA, more protein. Very good. What else? What other kind of things? Methylation of tumor suppressors, right? Tumor suppressors are not going to be expressed. So now you know exactly what they are going to do. So it is not surprising at all that epigenetics plays a very important role, and cancer also hijacks the epigenetic machinery. Okay, because those are the ones which are telling which ones to be expressed and which ones should not be expressed. Okay, so that's what it is telling you. So, so this is epigenetic problem. What you said is now shown here. So normal cells. So now you guys can follow too. Promoter region. Can you see promoter target gene? So this guy is methylated here. So methylation will close it, correct? So this will be, so for, yeah, so this is methylated. So then the target is not expressed as much. So in normal cells, oncogenes are off. But in cancer, No methylation. Now this is expressed in huge amount. Is it making sense to all of you? Now let's look in terms here. Okay. So in this case, in normal cells, it is partly methylated. So that means it is partially exposed and it is present in small amount, right? Partially exposed. But now, in this case, hypomethylation, barely any methylation, too open. A lot more gene is expressed here. Do you guys know what are exons and introns? You know? You don't know? That's okay. We'll discuss that later. Right now, just treat them as genes. I'll come to exons and introns later. Okay. Now here, in this case, let's assume this is a tumor suppressor. So in tumor suppressor, they will hypermethylate it. So the tumor suppressor is not expressed. Okay. And this is a normal circumstances. So, okay. So in this case, so every time you see here, right? So this is one gene, this is another gene, this is another gene. Okay. So these are three different genes. Okay. So don't worry about exon or intron right now. Just treat them as a gene here. And every gene has a promoter. Okay. So the promoter for this one is this one is this one is this, and for this, these are the promoter region. For this exon, you have this promoter. For this exon, you have this promoter. So look at the methylation level. So some are hypermethylated, some are hypomethylated. And accordingly, those genes which are hypermethylated are not going to be expressed. Those which are hypomethylated will be more expressed. So just in nutshell, oncogenes, hypomethylation. Tumor suppression, hypermethylation. Okay. So, so this is telling you about the clinical relevance of all different kinds of things. Whether your tumor suppressors are deleted, whether they are functional, you can check it by genetic testing. Yes, go ahead. So, so there are many DNA methylases. Okay, so some DNA methylases are overactive or less active, depending on that. So they have substrate specificity. So not all methylases will methylate all promoters. They will do in specific context. So those DNA methylases, if they are overproduced, which methylate or they are absent in so those DNA methylase enzymes, which hypermethylate, which methylate tumor suppressor promoters, if they are absent, no, if they are overexpressed, then they will hypermethylate those promoters, and those tumor suppressors will not be expressed, and that can lead to cancer. So basically, it is the expression level of DNA methylases themselves that can regulate. Second thing is that DNA methylation can only occur when there is some promoter exposed there, right? So there could be other mechanism where the promoter itself is not that exposed there. It is partially exposed, or there is something which is blocking the DNA methylase to come and hypermethylate. So it's a very, very complicated process. So it is not that all the promoters are exposed all the time, where DNA methylase can come and methylate it or not methylate it. So there could be some kind of blockage here also, which can prevent DNA methylase to come here and methylate this thing. So many factors can lead to change in epigenetics. Histones, yes. Repressed, yeah. Yes, yes, yes. Oh, that's so. Here I have only those slides which I brought it with me, so I don't have all the things, but I have really beautiful, beautiful pictures, in my lectures. So this is a very interesting thing, and I really wish I had those images here. So what happens is that, so you know the process by which DNA makes RNA. What is that process called? Transcription, right? Have you guys looked at the transcriptional machinery in humans? Probably not. Have you looked at it? So I am telling you a very simplified version. RNA polymerase makes RNA, which is 100% true, but it is a very complex process in the sense because DNA is so compact with all this chromosome kind of thing, right? So in human, the DNA is never open all the time, never ever. It just opens partially. So there are these many proteins they come together, many proteins they come together to keep the DNA, to expose the DNA. So what happens is that think of the DNA like it, just think for it that the DNA is extending from that wall to that wall here, right? So, and where I am standing, I am the promoter region, and this gene needs to be expressed, right? So you would think because, well, the DNA is extending up until that, but I am the promoter region, and this DNA is going that way. So you would think the whole show has to happen here because I am the promoter region, and these guys will have no say to that, right? Because this DNA is, we are part of the same thing. But this is the gene, this is the promoter region. So that DNA. But what happens in reality, because DNA is so tightly wrapped around histones and all of these things, there are proteins. What they will do, they will bind that part of the DNA. So on one end, so I am DNA, this is DNA, I am not a protein. But some protein will come, it will bind where that lectin is there, it will bind there, and then it will tilt the DNA and the other part will bind to this one here. So now it has distorted the DNA like this so that this part is more open. So what my point here is that there are hundreds of proteins which come together and bind different parts of the DNA to control the gene expression there. So if you delete something there, or if something is not binding, or if something is overexpressed, this gene will be more exposed, less exposed, or RNA polymerase. So RNA polymerase, so if this is the DNA sequence and this is RNA polymerase, it will not bind like this only. It has two legs. And these two legs are fixed legs. They are not like our legs that we can have two legs like this or this, right? We have some capability of moving, but it has two legs and it is fixed. So when it sits on the promoter region, this is fixed length. It's not like movable length. So it recognizes two parts of the promoter where its legs are touching it, right? So if the promoter sequences are changed at two parts, it is not going to bind very nicely. So remember I just said a few minutes ago that promoters are strong or weak. So intentionally. So some promoters are strong, RNA polymerase will bind very tightly, very happily, it will make tons of RNA. Some promoters are weak, it's wobbly, okay? It binds but not very happy, less RNA, right? So the main thing is that it is intentional. The transcription is not the same for all the genes. Some genes make a lot more RNA, lot more protein. Some genes make a lot less RNA and less protein, okay? And there are many proteins which come together, and one of the mechanism is, so epigenetics we are talking about. So suppose it is methylated, then many proteins will come and bind the methylated part. So remember I showed you here for DNA. Now here it is for histone. When it is acetylated, nothing binds here, more open. See how open it is now. But when it is methylated, these HP1 proteins come and bind like this and completely block access to the DNA. So acetylation can also recruit transcriptional machinery to help in transcription. So it's not binding this thing. It is actually binding to those proteins which aid in transcription. Makes sense? So keep in mind, I'm showing you the simple version of everything, okay? Most of the time acetylation opens up, but once in a while acetylation can also inhibit it. But I'm just telling you what happens most often, okay? All right. So now go ahead. Yeah, go ahead, go ahead, go ahead. They have NH2. I have never seen methylation there. It's mainly limited to arginine and lysine. I have never come across glutamine or any other amino acid besides these two that are methylated. And lysine methylation is most common. Okay, so now you guys already understand precision medicine. Precision medicine is that you find out exactly what went wrong with this particular patient and then you treat that problem. So suppose in many cases, the main reason for cancer is aurora A kinase is deregulated. So you don't have to give chemotherapy, you just give aurora A inhibitor to that patient, right? And that will be very good. It is besides the point that aurora inhibitor is also very toxic to start with. It is almost as worse as chemotherapy because aurora is expressed in every cell of our body. And if you inhibit aurora A, every cell of our body suffers the consequence. But the point here is that if you figure out the exact mechanism, you can give that disease, you can give that therapy. So that is personalized medicine or precision medicine. So patient A has this mutation, so you give this drug. Patient B has this mutation, you give a different drug to that. That is precision medicine, right? So that is what it is telling you, that you don't, it is not one size fits all. Every patient is unique and every patient has to be given accordingly. See, many cancers which are very bad, they are caused by one major player. And if you find that major player, you just give that drug. Okay? So this is now I'm telling you specific case study. So this is breast cancer. So a lot of breast cancer, they overexpress a receptor called HER2. Okay, so HER2 is also present in normal cells, and it is a growth factor. So it gives the signal to cells to divide, and when it is turned off, it is turned off. But in cancer, it is overexpressed. Okay, so because there are too many of them, too much signal is coming to the cells, and these cells just keep on growing all the time. So we know what is the culprit. HER2 is the culprit, right? Once we know, so this is one of the most famous drug. My friend took this drug, Herceptin. Her breast cancer is completely cured. She had breast cancer when she was, I don't know, she was in her 20s, and she was treated, and now she's very, for decades, no cancer. It never came back. So it's a very effective medicine. Okay, so Herceptin is an antibody against this HER2. So if you give Herceptin, no signaling. It just binds, keep them apart, and it is very effective. Breast cancer is one of those cancer which is very easy to treat unless it is triple negative. Okay, so breast cancer express 3 different kinds of receptors. So if it is a receptor associated disease, it's easy to treat because you give the antibody. Triple negative means all the 3 receptors are gone. And then how do you give an antibody? So that is the one which is very hard to treat. Okay. So this is lung cancer. So EGFR is also a receptor which belongs to the HER2 family. So what happens is that under normal cell conditions, when EGFR is so do you know what is a ligand versus the receptor? Do you guys know that? So do you know chemistry background? Probably not. Okay, you know. So let me still say it one time. This is a receptor, and this is so this receptor is poking out of the cell surface, right? So if this binds the ligand, this is the ligand. If it binds the ligand, it is turned on. As long as the ligand is not there, it will not turn on. Most of the times, these receptors are like this, right? And when it binds the ligand is like this. Okay, so it brings 2 receptors close, and when they are close, they phosphorylate each other and they are turned on. So in many cases, when they are separate, no signal. But this ligand is the one which will bring two receptors together. They dimerize, and once they dimerize, that's when signal is turned on. But when antibody binds, it binds separately. They will never come close together because now the antibody is blocking the binding of the ligand, so they stay apart, signal is off, right? So this is showing you different ways EGFR receptor is activated, especially in cancer. So under normal circumstances, they are separate. When EGF comes, two receptors come, signaling is turned on. EGF is removed, signaling is turned off. Okay. But here what is happening, there is some mutation in there that even these are super acceptors. So super acceptors, binding. EGF, when it binds, and if you have very little EGF and EGF binding is poor, then you will have less dimerization. But yesterday we were discussing, I don't know who was talking to me about prostate cancer and androgen signaling. I spoke to so many students. Somebody was talking to me about prostate cancer and androgen signaling. So one of the things they do, they increase their affinity towards the ligand. So here, even if you have less EGF, they are super binders, super acceptors. So even with less EGF, they will dimerize and turn on the signaling. That's bad. Okay, here, so remember I said two receptors come together, they phosphorylate each other. So here, they are always bound to ATP. This part is super active. So even if it doesn't dimerize properly, this part is already active and it is turning on the signal. So that is bad. And here, this is the extreme case. They come together even in the absence of EGF. They dimerize without EGF. So they are always turned on. They don't even need the outside signal. Okay, so what is the cure for that? You make, so many times the receptors, this pocket is like this under normal circumstances, but the pocket is mutated in cancer. So if you make an antibody which will recognize this pocket and not this pocket, so normal cells are unaffected, but the cancerous cells which are expressing this kind of one, they are the one which will be shut off. So this is what is mutate specific antibody for cancer only. Okay, in this case, suppose it is a normal EGFR in cancer, but it is present in too much amount, then you can make an antibody and it will bind those and it will shut it off. Okay. Third case, CML. This is I wanted to tell you for a very long time. Okay. How many of you have heard of Gleevec? No one. So there is no chance that you have heard of this protein called BCR-ABL. So why Gleevec is so famous, and I'm part of that story. So Gleevec was the first tyrosine kinase inhibitor which got approved, and it got approved by Novartis. Okay, so and I was working at Novartis, and I was working in the kinase field. Having said all of that, I have nothing to do with Gleevec. But I was there when Gleevec was approved. So I was part of the celebration of Gleevec by Novartis. So I enjoyed all the perks without contributing to anything towards Gleevec. Okay, so we were flown first class to Mallorca in Spain, which is a very, very beautiful place on the Mediterranean Sea. And, we took a ship and that sailed on Mediterranean for almost a day. And throughout the day, we were served snacks. So they have a special word called tapas or something. I can't remember, but it's a snack on a stick. And all throughout the day, they were changing the snack. And it was huge celebration on the Mediterranean for Gleevec. And I did not do anything, but I was just there at the right time. So I had all the fun that originated from Gleevec. So it's my favorite story. Okay, so what happened and how it became a billion-dollar drug and saved the lives of millions of people. So what happens is that there is a kinase called ABL. Okay, so in normal cases, it is off and it is turned off for a very short period of time and then it is turned off, right? So this is ABL. So this is inactive. So this is the catalytic domain which accepts ATP and phosphorylates protein. and this is off. So this is the whole protein. This is not different protein, one protein, different parts. Like this is head, this is torso, these are legs, right? So this is the enzymatic side, this is other domains, and the tail binds to this and keeps it off. And it is turned on for a short time, then it is turned on. What happens in CML, chronic myeloid leukemia, these patients have gene translocation. Remember, there was gene translocation. I said I'll tell you later on. So what happens is that one part of the other chromosome migrates and binds. This is ABL gene. So it migrates and comes here. I have another slide also I'll show you. So because of this fusion, ABL is a small protein, but this BCR from this thing translocates here. Now it becomes BCR ABL. And when it is in the BCR ABL form, it's always active. And that leads to CML. It's a very bad disease. Okay. Oh, this picture is already here. So see, ABL is this blue part here. Okay. And in BCR ABL, another chromosome, this BCR part comes from different chromosomes. So this is chromosome 22. ABL is in chromosome 9. But there is a chromosomal break where this part comes and fuses with this part, and it makes a huge protein. This is 220 kilodalton protein. And when this protein is expressed, the catalytic domain here is always, look at this thing. This is always, always, always on. And that leads to cancer CML. So this drug Gleevec, right? This Gleevec, so that binds exactly in the catalytic domain and shuts it off. And basically ATP can no longer bind and it is rendered ineffective. Okay, so this is how it works. And the other famous name for Gleevec, initially it was called imatinib, but the commercial name for patients who buy, that's Gleevec. So that is that story. So in many cases, something becomes hyperactive, and if you can design a drug which specifically binds that hyperactive protein, you shut it off. And another story about Gleevec is that, Gleevec was very effective for BCR-ABL. So many companies made inhibitor which was even tighter binding than Gleevec, but it was not very effective in patients. So that's when it was realized that Gleevec not only binds BCR-ABL, it binds PDGF receptor, c-KIT, and a few others, and they are all deregulated in CML. So that is why Gleevec is hitting all the bad targets in cancer, along with BCR-ABL, and that is why it is a relatively dirty drug, but all the correct targets, and that is why it is so effective in CML. But guess what? So when Gleevec was given to patient, it was fantastic. A large percentage of patients responded, and it was so much celebration. But then few patients in no time stopped responding. So then it was checked why they are not responding. So their gene was isolated BCR-ABL. It underwent mutation in the active site. Now Gleevec no longer binds there. So that's the problem. They develop resistance because they undergo mutation. So this is the worst part of cancer. So I always tell when somebody is going chemotherapy, you better get it cured fast. I mean, it's not in someone's hand, it's not in my hand or that person's hand. But if the therapy continues for a long, long time, then the chances are cancer will develop resistance. That's why many people get well and then cancer comes back. That's the main reason, because cancer undergoes mutation and they become resistant. I'll stop here, right? Good timing? Okay. Okay, 15 minutes break for us. Today we are having 4 lectures, right? That's what we decided. Okay. We have to compensate for the break the first day. You guys were forced to take a break. I was also forced to give you a break because I was stuck at the airport. All right, see you guys in 10-15 minutes. 15 minutes exactly. 12, 10 minutes. So what's the schedule today? So 12 to 1 we have one more class, then we have dinner until 2.
Lunch.
And lunch until 2. And then we have tutorial until 3:30.
So maybe from 2 to 3 you can take one lecture, madam. So that one lecture will be compensated.
Oh, one lecture. Of course, one lecture. Yeah, I forgot.
So
This
2
is
to
the
3
third
we'll have
lecture.
one lecture, then break for 15 minutes, then tutorial.
Yeah,
From
so
3:30 to 5:30 we'll have the tutorial.
okay, that sounds good.
So slowly in 3 days we'll compensate.
We'll have intense 3 days. Then we'll have less intense remaining days. And then we will have Sundays in between to recover from all the tiredness.
Someday we want to keep a holiday for that.
Would you prefer? Yeah, Sundays we keep holidays. We all need break. No matter what, we need break. I need break so that I can visit nearby places.
So Gleevec has become resistant to many patients or only
Yeah,
very few?
even after 6 months, when Gleevec was first time given in 2001 too, this resistance emerged within few months of the treatment. So, and that is always the case.
So they identified and they again changed it, madam. They
Yeah,
inhibited.
yeah. So now they are dasatinib. These are all second, third generation imatinib. So this is imatinib,
Yes.
and now the most famous one is dasatinib, where they have modified imatinib.
So that is where the computational will help.
Will help tremendously. So you are going to help tremendously when mutation occurs, how to change the molecule. So that's what you were asking. Who was asking me computational yesterday? You were, yeah, see? So now this is where this is going to
After
help.
developing a drug.
After developing the drug.
I will take note of that experimentation. Now, actually, in BCR, I guess there are 5 different types of mutations, like besides in the catalytic side.
Oh, there are many mutations. I was just telling you the simplest
I
version.
got confused when you said Gleevec, but like I heard about imatinib, like
Imatinib is Gleevec. It's the same thing. Yeah, it's the commercial name.
brand name.
Yeah. We used to call it BCR-able inhibitor at that
But
time.
we know it as like tinib, like
Imatinib, dasatinib, and all of these things. Yeah, that's the more common name.
I guess this is tuberculosis and gastrointestinal.
Yeah, GIST. Yeah, it is also because BCR-able is there also. Yeah.
One more thing, yesterday you were telling about genomic instability, then you talked about BRCA mutation and PARP inhibitors is also written there. So like in case PARP inhibitors are like they have PARP and many isoforms. So they are
I'm going to
non-selective.
tell about this today.
Okay, how does
I have
okay,
that.
then I will ask you that.
Yeah. I have it there. There is a lot of things there.
I have read about this olaparib.
Olaparib, yeah.
But
That.
you get more confused when it comes to the repair pathways for biology, like base excision, nuclear excision, then homologous recombination, then non-homologous
The
recombination.
thing is that that field itself is a whole subject in itself. So I have to be very brief because otherwise I will just go off track. So first of all are that what are these resistant mechanism and how do we counteract these resistance mechanism, and what is the Achilles heels here? So when cancer cells become resistant to some mechanisms, they develop an alternative mechanism, right? And that is why they can survive those conditions. But when they develop that alternate mechanism, there could be something vulnerable about that alternate mechanism. And if you can identify that kind of vulnerability, you can attack that weak part and you can kill that. So the normal cells don't have that new path, but this new path may also have its own weakness. And if you can identify that weakness, then you can attack that weakness. And that is what is meant by Achilles heel here. So there are two kinds of resistance in cancer. There is intrinsic resistance, and then there is acquired resistance. So suppose you develop a drug, right? And you give it to two different patients. One patient responds to that drug, the other patient does not respond to that drug. So one of the reason is that the other patient who is not responding to that drug, he is resistant to that drug. So there is something internally present in that patient which makes him or her resistant to the drug. So that is intrinsic. The tumor itself is resistant to start with, even before the treatment. Then there is acquired resistance. You keep on giving the drug, and like I was saying for Gleevec, those patients then develop a resistance mechanism. Okay, so that is acquired resistance. So for example, when you have intrinsic resistance, one of the reasons why this is always present there, why they are not responding in the first place, because they don't even have that target. Suppose you are giving against BCR-ABL, and if it doesn't have BCR-ABL, it's not going to respond to Gleevec. Or there is some mutation in the BCR-ABL in this patient so that it does not bind Gleevec. So again, so it is low affinity. It does not even binding that. Or they have some mechanism so that the drug is not even getting inside the cell. So this cell is not letting the drug come to that. Or there is something above the cell which is blocking access of that medicine or that drug to come inside the cell. Or there are many receptors here which throw the drug out. So what happens when you give a drug, and this is the body's defense mechanism also sometimes, that there are these receptors, efflux receptors. So you give something, they just throw it out. You give it, and so initially, let's say there are two of these things, right, in a cell. But when you are exposing them to the cells of now, many of these things will come and they will just throw it outside the body. So that is called efflux mechanism. They throw the drug out. So this is a very common thing. When you give drugs to the patient, these guys are going to be upregulated and they will throw the drug out of the body, out of the cell. So that is intrinsic resistance. And this efflux mechanism could be due to acquired also. It wasn't there initially, but when they got exposed to the drug for a long time, now they start throwing the drug out of the cell. Okay, so that is why efflux mechanism is also here. Okay, acquired is that initially Gleevec was working beautifully, BCR-ABL, but then these patients, after just 3 months of Gleevec, they develop resistance because now BCR-ABL mutated, now Gleevec doesn't bind. Okay, so or they have developed new mechanism that they don't even depend on the first target. So they have alternate. So suppose I have this mechanism, you block this mechanism, I develop this mechanism. So suppose you are buying your chemical from Sigma. Sigma stops selling it, you buy through someone else, right? So another pathway, okay? Or the target itself is modified. So there are many ways to acquire resistance. So this is intrinsic resistance, intrinsic mutation. It was there from the start. And so initially when you gave the drug, most of the cells die, but these guys which were resistant because they already had that mutation, these guys will make a new tumor. Now you can keep on adding the drug, nothing will happen. Here, these guys were pretty naive. You gave it, some are survivors, right? We know some are survivors. So they changed themselves to survive the conditions, right? And now they make the treatment. They are resistant. Okay, so key drivers. So the problem of resistance. So genetic mutations occurs. The protein modifies itself. We introduce new mutations so the drug cannot bind there. Epigenetics rechanges, right? They will change the DNA methylation pattern, histone acetylation, deacetylation pattern, and they will activate new genes and things like that. So they will rewire the system. Okay, tumor microenvironment support. So once again, let me remind you about the tumor microenvironment, and I'm going to discuss a lot about this thing. This is a relatively new field. So this is cancer tumor. This is the tumor, right? So this tumor alone is not responsible for cancer, right? This is, you can think of tumor as a very, very, very super smart guy. So this is the tumor and this is going against the body, right? So it is so smart. If it encounters some drug, it will mutate to protect itself. It will steal from other guys to survive because it is growing so fast. So there is a lot of stealing going on from normal cells. It fools the immune system. It acts and it tells the immune system, I am just part of you, don't kill me. Okay, and then it will recruit all the things to shut down the immune system. So this is the tumor, but a lot of it is going outside the tumor. All the environment outside is supportive to the tumor, and it will send off all the bad signals from outside. It will protect itself from many different things. So that all forms the tumor microenvironment. So immune system helps us, right? You know that. So there are many cells which are considered anti-tumorigenic. So if they come, they will kill the tumor. So tumor cells will secrete something and make anti-tumorigenic cells pro-tumorigenic. Now they have changed to help them. So not only it is like you take the enemy and you make it your friend. So it has actually come to kill the tumor. Now it is helping the tumor to grow even bigger. So that is all because of change in the tumor microenvironment. One of the things they do, they secrete a lot of acid. So the whole environment is acidic. And in this acidic environment, nothing can come close to that, which can kill the tumor. Okay, and then if you make too much acidity, things will break apart, tumor can spread around. Okay, so that is all comes under tumor microenvironment. And phenotypic plasticity, that suppose somebody is considered a culprit in one country and that person changes the name and completely hides itself and he's or she's living in the society but nobody knows. So that is phenotypic plasticity, just completely change your appearance. And then the drugs cannot recognize you because you have changed. You don't depend on the protein that the drug is against. That is phenotypic plasticity. Okay? So let's discuss a little bit of many different mechanisms of resistance. So genetic mutation, you have changed the target itself. Drug will no longer bind there. Gene amplification, now you make a lot more protein than before. So initially you were making this much protein, the drug was effective. Now you're making so much protein, the drug cannot even compete with that. Okay, you can bypass that. Suppose the drug was, suppose this cancer was due to something. So you give so like prostate cancer depends on androgen receptor signaling. If you give androgen receptor signaling inhibitors, after some time they don't even care about androgen receptor at all. They completely forget about androgen receptor. They don't express androgen receptor. They don't depend on androgen. They don't care about it. There is no androgen related signaling there. Now they overexpress MYC protein, and MYC is the one which is leading to cancer. You can keep on adding androgen inhibitors, who cares? They don't need that anymore. So they just bypass the whole signaling pathway. This is the bad way. They overexpress efflux prem. So you add drug, they throw it out. You add drug, they throw it out. Okay? This is another thing. So whenever you think of a tumor cells are not all the same cells. there is huge heterogeneity. That means that there are many different things which are wrong in this tumor. So suppose you're giving drug against that. Okay, it will kill this part of the thing because this part of the thing depended on that target. But there are many different targets in different parts of the tumor. So tumor is so heterogeneous. Even if you kill one part of it, it's fine. The rest of it will still grow and keep dividing, and the tumor will keep on growing because the tumor is really heterogeneous, right? And then, so let's do radiation therapy. We give so much radiation, will damage the DNA, tumor should disappear. Now they will upregulate DNA repair enzymes, tumor cells. You give them damage, they repair it. And you know what? If they inefficiently repair it, no problem. They will make more mutations, adapt, move on, right? So, and then they will do epigenetic rewiring. Again, oncogenes are overexpressed, tumor suppresses down. So global hypermeasure. So they can do a lot of things. They will inactivate tumor suppressor. They will activate genomic thing. So they can do all this epigenetic rewiring to support cancer. Okay, and keep in mind, you know the survival of the fittest. So even if some die, who cares? Those who die, they die. You are not meant to be part of the cancer thing. I'm just telling you cancer philosophy. Those who survive are the ones which will make more. So whoever survive, even if there is one single cell, plenty, that one single cell will form the whole tumor, that one single cell will metastasize, right? So if they're dying, nobody cares about dying cells, because that's why I was saying slowing is sometimes the best option. But if you can kill it, that is absolutely the best option. But cancer can be so devastating that slowing itself is considered really a big win, right? Especially when some cancer comes unannounced and the person doesn't know anything and they think, they have 20, 30, 40 years to live, and then cancer shows up. So then even if you can extend life by 6 months, it's a win, as long as the quality of life has not deteriorated too much. Okay, so now let's look at the tumor environment. So look at this part here, okay? So what this is telling you, that there are many different kinds of cells which are present in tumor microenvironment. This is a picture I generated using AI. So AI always messes up the picture, but it was still beautiful enough for me to put it here. Okay, so here you can see look at all of these fibrous structures. This is not ideal, but basically a tumor is surrounded by hundreds of different kinds of cells which support the tumor growth. Okay, and those are extremely important for protecting the tumor. So if you can actually attack the tumor microenvironment itself, you can reduce cancer. Okay, so now let's talk about strategies to counteract these resistance. So, so first you figure out exactly what is the cause of cancer. Okay, this is very important to figure out what is the cause of cancer before you make your plan of attack. So for example, like I was telling you yesterday, in pancreatic cancer is the most lethal cancer. So pancreatic cancer does not occur in many patients. So if you take, I don't remember the number, in terms of cases, pancreatic cancer is maybe the 10th it is 10th in number, as I recall. So not many patients get pancreatic cancer, but if they get pancreatic cancer, it is the leading number one cause for cancer-related death. So most of the patients who get pancreatic cancer do not survive. Less than 5% of people have a survival rate for more than 5 years. Breast cancer, it could be 80% people survive, 85% people survive. For pancreatic cancer, it's less than 5%. It's the most lethal cancer. So this is the tumor, and this big is the microenvironment, this big. So they are surrounded by a lot of other cells which protect the tumor. So what people thought, that they poke hole in these whole fibrous mess, and they can get the drug out to the pancreatic cancer cell and it will die. and you can treat it. But the problem is that this is what I meant by you have to have a plan of action before you attack cancer. So if you poke hole, right? If you break the fibrous thing to get the drug inside, what is the side effect? Now you are making it a channel for pancreatic cancer cells to escape and form tumor elsewhere. So it's not a good idea to break also that, because so far it is more contained because of this whole fibrous thing. But breaking open it so that it dies can also lead to metastasis. So you have to have a very solid plan to attack cancer. Okay, so that's what it is saying, that you have to really figure out using genomics, proteomics, metabolomics, all the tools that we have to find the real cause for that cancer, and then you can decide exactly what kind of therapy you are going to use. Okay, I know of someone who was diagnosed of very aggressive cancer recently, and by the time it was diagnosed, it had already spread to other organs. So it was imperative that before going and really figuring it out, which would take at least a month or two, and the cancer was spreading so fast that patient didn't have 1 month or two just to figure out the thing. So what they did before figuring out exactly what went on, they started immunotherapy because they wanted to contain the disease. And now that person is responding to immunotherapy, right? And the disease has stabilized, and it is also regressing a little bit. So sometimes you have to make a decision to make it stabilized. Don't let it spread. But at the same time, you should also make a priority to figuring out exactly what kind of precision medicine is needed. Okay. So cancer cells undergo a lot of things to have all these bypass mechanisms to avoid that. So how can we identify the weakness of cancer? So in ideal situation, normal cells should not be affected by the disease. Cancer cells should be affected by the medicine, right? So that is the whole point. So I have already told you about the resistance mechanism, but I will tell you how do you overcome drug resistance. So we can sequence the DNA to find out what kind of mutation it is. We can use CRISPR-Cas9 to change the DNA. These things are not easy, but these things are being used. Okay, so we can change the DNA. We can do high throughput screening to find the drugs. Somebody, you were asking me, right? Whether we should do molecular modeling first. So the way pharmaceutical industries, they identify drugs towards target, they have so many. See, in our academic labs, we make 10 molecules or 20 molecules or 50 molecules. And it's a huge amount of work to make 50 molecules, 50 different molecules. But industry, they already have millions of compound in their library. So they do high throughput drug screening because they already have compounds in hands. And then they see, okay, these are the hits. And then they start from those hits. And then they modify the hit to make it even more potent compounds. So you can do high throughput screening to identify. You can do combination in high throughput screening and see what two drugs in combination can be more effective. So this can give you results in weeks, right? Combination. But then obviously you have to test it and everything. Okay. Now, this is something I have never talked about. This is biomarkers. So we have a whole lecture. Probably tomorrow I'm going to tell you about biomarkers in details. But biomarkers are basically will tell you what kind of therapy will be good. So for example, remember I told you about HER2. So HER2 is a biomarker for that cancer. If that patient is expressing HER2, you know we have to give Herceptin as a disease because Herceptin is an antibody which will bind HER2. So your biomarker is telling you what therapy you should use. Sometimes the biomarker can tell you whether the drug is going to be effective, how much effective, whether you should change the dose. All of these things come under biomarkers because there are many, many different kinds of biomarkers. So biomarkers really help in precision therapy. Okay, So, and then we can combine it with proteomics, genomics, immunotherapy for cancer. Okay, so these are few different ways you can tackle cancer. So instead of giving one drug, you give combination of drugs. So, do you know what is synergistic and additive effects? Some of you know. Do you know? All of you? Probably not. So suppose you give A drug and B drug. A is working, B is also working. But if you combine them together, so this can be additive. If this was 5%, this was 5%, you combine them, now it becomes 10%. So 5 plus 5, 10. Additive effect, right? Or when you combine this and this is 5% alone, this is 5% alone, but when you combine them, this becomes 50%, synergistic. So this is not additive, it is synergistic. So it is much more than the additive effect, that is synergistic. This is ideal situation to combine them, and there are many cases where you use combination therapy. Or sometimes this is 5%, you combine it with this is 5%, now it is 2%. That means these two cannot work in combination. They are basically killing each other, right? You don't want that. So combination therapy, you have to figure out what combination. Sometimes it could be a combination of 3 drugs. I know cancer patients are usually given 2 kind of therapies together, angiogenesis inhibitors and immunotherapy. And that is very effective, okay? Because you are stopping the progression by inhibiting angiogenesis and you are also treating the cancer. So 2-prong attack is very effective. Okay, so that is combination therapy. Immunotherapy is very effective. This is a game changer in cancer field, immunotherapy. And I briefly told you about PD-L1 therapy yesterday and CTL4. We'll come back to that more when I tell you about immunotherapy. Okay, tumor vaccine. So here we were discussing vaccine during the break, right? So if you take tumor specific protein and you make a vaccine against the tumor specific thing, normal cells will not be affected, tumor cells will die. Okay, then gene therapy. If tumor suppressors are defective, you introduce a gene to express tumor suppressors. That would be a gene therapy. If you have an oncogene, you can send an shRNA to degrade the oncogene, right? To degrade the mRNA of the oncogene, you basically are removing that. Either you can use an inhibitor to shut down the protein, or you can just get rid of the protein altogether. So somewhere, I don't know which day, because we are going at a rapid speed, but I'll tell you about PROTACs. And in PROTACs, you make the protein disappear. Okay, but in this one, it is showing you that you can degrade the RNA. Antibody drug conjugate. So antibodies, this is very powerful method, antibody drug conjugate. So what you can do, what is the feature of antibody? So suppose this is protein A and you make an antibody, and you can have millions of other protein, but this antibody will only bind this protein A. This is so specific. Okay, so suppose you make an antibody towards a protein, but you also hide the drug in the antibody. So now the antibody will go to the tumor, bind, and the drug will be delivered here, and this tumor will die. But you don't know what drug to deliver. You attach radiation therapy, it will directly go to the tumor. Or you can attach an imaging agent and the tumor will light up. So even if you have a single cell, that single cell will show in the imaging because you have light up the tumor. So that is antibody conjugated drug, ADC, right? So people attach all sorts of drug and deliver it directly to the tumor. Okay, then targeted therapy like HER2 therapy I just showed you, right? Receptors overexpressed, you target those ones. So this is target. BCR-ABL, targeted therapy. We are not giving chemotherapy, we are just giving the drug. BCR-ABL is not present in normal cells, so you are actually only giving it to cancer cells. That is targeted therapy. Okay, you can introduce different kind of cell death to tumors. Okay, this is very important. CSC and EMT. CSC is cancer stem cells and EMT is epithelial to mesenchymal transition. So think of normal cells like bricks, right? So these normal cells, they look like, these are cancer cells, but they look like this, right? They're pretty, wide and everything, more organized. So they cannot go through the blood vessels easily because these cells are not very mobile. So they undergo a process which is called epithelial to mesenchymal transition. So these cells, they become more mesenchymal. So if this is the normal cells, I also have a slide to show you this thing. They will become like this kind of cells. Look at the shape here, really different. So this is epithelial morphology and this is mesenchymal. Okay, so what is the advantage of this transition, epithelial to mesenchymal? Mesenchymal, they are very narrow, so they can go through this hole and they can metastasize, they can get into the blood vessel. Plus, they're very motile. Epithelial are not very motile. Mesenchymal is very motile, so it can squeeze in through the space, go out and metastasize in other organs. And once they reach some other organ, they will become epithelial again and expand the colony to form a new tumor. So that is EMT. When EMT occurs, cancer is considered really bad. And cancer stem cell, so remember, stem cell can differentiate to any cells. So when cancer, so what are cancer stem cells? So suppose this is the tumor, right? And you give radiation therapy or any therapy and you kill 99.99999% of tumor cells, but literally one cell survives. Just one cell survives out of the trillions of cancer cells. That one cell is resistant to radiation, resistant to drug, resistant to that, and that one cell is the cancer stem cell, which is just resistance to everything, and that can make a whole new tumor. And those cells are very hard to kill. But now we also know how to kill those cells because we know what exactly goes inside those cells and which proteins make them so resistant. So attack those proteins, even those cells will die. Okay. And lipid nanoparticle is just a way to deliver the cargo to the right location. Okay. So let's talk about different ways. So I'm going to tell you about 5 different things. Inhibitors. So like Gleevec was very effective. Resistance emerged, then they make second generation, third generation. And one of the very common one, now nobody uses Gleevec, they use Dasatinib. Gleevec was called Imatinib, and then Imatinib was converted, same scaffold, same thing, new changes around the same molecule to counteract the resistance. So Dasatinib is the next generation inhibitor, and this is even more effective than Imatinib, Gleevec. Okay, so you can make more and more modifications. So these are called next generation inhibitors. So first generation inhibitors you modify, second generation you modify them, third generation. So cancer is modifying, and so we are modifying the molecules accordingly, right? Combination therapies, synthetic lethality, PARP inhibitors, we'll tell about that. You will love the synthetic lethality section, and then we'll talk about tumor microenvironment and epigenetic modifiers. So next generation is very simple. You give the drug, resistance emerges, and then you modify the compound. Okay, so that is, so what is the characteristic of better inhibitors? They have better selectivity, right? So when you guys are making inhibitors towards it, right, it is very easy to identify inhibitors. The main thing is that it is only hit your target and no other target. So selectivity is more important because oftentimes if it is non-selective, it is also hitting normal cells because it is binding other targets, right? If there is resistance, that the inhibitor should be modified so that it can overcome resistance. Improved safety, it should not be toxic, right? If it is selective, it is going to be less toxic. And allosteric inhibitions. So what happens, especially in the kinase field, all kinases have ATP binding pocket. So many kinases, the pocket looks very similar to each other. because they all bind ATP. But they all have allosteric pockets, like pockets outside of the catalytic domain. So if you make something which will bind allosteric site, those are very unique to kinases. So those inhibitors tend to be more selective. So sometimes instead of targeting the active site, you can target other pockets. Do you guys know what is allosteric inhibition versus competitive inhibition? Everyone? You do not so clear about it, right? You guys appear apologetic, but I'm not teaching chemistry, because then these guys will be asking me the same question. So ask me anytime, okay? All the time, if it is not clear. So what happens in allosteric, that this is the catalytic site, right? And imagine this whole hand is the protein. So what happens is that something is here, there is a pocket here, and if something binds here, this closes. Okay, and something binds here, this opens up. But this two part is not part of this. They are very far apart. But binding here or here is controlling whether it is turning on or off, isn't it? So that is allosteric. It's not in the active site, it is further away, but this binding is changing the conformation of the enzymatic active site. Is it making sense? So that is allosteric. So you can make inhibitors two ways. You can directly block this, right? That is competitive inhibition, because that's where the substrate bind, you are competing with the substrate. But if you bind here or here, that is allosteric. Very simple words, right? So allosteric sites tend to be different from one protein to the other. So you can achieve more selectivity. That's the point here. Sometimes allosteric sites can be deep buried in the pocket. So they are not directly controlling that, but if you target that pocket within the active site, you can achieve higher selectivity. That's what it means. Okay, simple words. So these next generation inhibitors, they target these specific things, right? So all your classroom, they look the same, but this classroom may have something special there. So you can make use of that special thing. So most of them are rectangular classrooms, right? They have the same shape, but every classroom, even in this building, has something unique about it, right? And that unique, it is still the classroom. When you're sitting here, you're not accessing that pocket, but this active site still has that special thing there. That's allosteric site, right? So that site is not helping all of you sit here, right? So if ATP is coming here, that allosteric site is not really helping ATP, but it has the uniqueness hidden there. That's the allosteric part. Okay? Okay. So this is just showing you, next generation inhibitors, and it overcomes. So this is third generation, fourth generation. So every time cancer mutates, we also change the inhibitors. So we keep on calling them next generation inhibitors. Okay, now let's look at combination therapies. Okay, so in combination therapy, it could be additive, like 1 + 1, 2. So this is 5%, this is 5%, you combine, it becomes 10%. Great, better than 5%. Okay, it could be independent, so that these two drugs then don't work in the same pathway, but if you use them together, it's like immunotherapy. I was telling people are given immunotherapy and they're giving VEGF inhibitors. VEGF means angiogenesis inhibitors. So they are two different things. Immunotherapy is activating the immune system. This is stopping the growth of new blood vessels, totally independent. But both are killing cancer. Okay, and then synergistic. This is best. 5 + 5 together is 50%. That's synergistic. That is very, very good. Okay, and antagonism, once in a while, many times if you don't combine them together, it is very bad for cancer. The drugs themselves kill each other. Okay, so advantages of combination therapy, of course, if you use the right combination, it is more efficacious. Okay, you can kill cancer fast, you can overcome drug resistance. Okay, so now they are trying to develop drugs which is like you just hit cancer cells with a brick load of things. Actually, brick loads of things. Imagine a truck with bricks, and you come and just you dump all the bricks all together. Like in no time, the whole brick is given there. They are going to collapse with the weight of those bricks. So it's like the whole, and these are actual that kind of thing. So they assemble a lot of things together, right? It's like a huge thing. And then cancer cells express the enzyme, which is only specific to cancer cells. And that just lights the fire here, and the whole thing collapses like this, and the cancer cells just die right away. Okay. So then they don't develop resistance, because they can develop resistance if they have some time to develop resistance. But if you attack them so suddenly out of nowhere, the whole thing dies. Okay, you target multiple population. Remember, tumor is heterogeneous. If you inhibit one thing, the tumor may not regress. So if you're attacking from multiple directions using combination therapy, you can kill the tumor quickly. If you use the right combination of drugs, it may be less toxic. If you use the wrong combination, it will be very toxic. Okay, and this has improved patient outcomes. Okay, what are the challenges? You have to choose the drugs very carefully. The drug should not interact with the drug itself and kill each other. So drug-drug interaction, and the more drugs you use, there is a chance of more toxicity, more complexity. Okay, and like we were talking about vaccine, like you give vaccine for the right cause, but it can also cause its own thing. Okay, I'm a pro-vaccine person, but there is always a possibility that vaccine itself will introduce more complexity in some patients. Okay, and if you use more, it is going to be more costly, right? So that itself could become an issue. And then again, every person is different. Most of the people are benefited from vaccine, but there are always some patients, some percentage of patients who develop long-term complications with even as simple as vaccines. Okay, so type of combination therapy. So these are very common type of combination therapy, that people use chemotherapy and radiation together just to kill the cancer cells. Okay, immunotherapy is often used with angiogenesis inhibitors. Immunotherapy can also be used with chemotherapy. Immunotherapy can also be used, it doesn't show you here, but it is used with many other combinations. So basically, to kill the cancer cells. Targeted therapy, sometimes you use two targets at once. Sometimes what happens is that you are inhibiting this target, but you are also inhibiting the target which is below that. So if this escapes that, at least you kill this. So if you kill two in the same pathway, the chances are the whole pathway will shut down. Okay, chemotherapy and immunotherapy are mixed together. Chemotherapy and angiogenesis therapies are mixed together. Radiation and immunotherapies are mixed together. So these are different kinds of combination therapy, but in combination, remember yesterday I also said sometimes before before undergoing surgery, people give chemotherapy to reduce the tumor before surgery. Sometimes they do the surgery, then give chemotherapy. If something has escaped, they will die. So they combine different kinds of therapies. So radiation therapy, chemotherapy, immunotherapy, targeted therapy, they are often used in combination. Okay. So rationale for combination therapy, you are attacking cancer from multiple directions. Okay. Now we come to very interesting topic called synthetic lethality. So what exactly is synthetic lethality? So in very simple words, so do you have a class? Oh, you are for this class. Okay. Usually when you teach a class and students come towards the end, you think that there is a new class and I have to wrap up this class. So without thinking, I had the same reaction, because when I'm teaching class, students start to accumulate outside and I know my time is up. So you elicited the same response in me. that's fine. I didn't want to corner you there, but like years of teaching and students sitting outside, it's a sign. Wrap it up. Okay. I forget, what time did we start?
Yeah, 45, so 1 o'clock.
1 o'clock, plenty of time. Okay, so this is a very interesting topic. So our cells are very smart. You shut down one pathway, they will do the same thing using the second pathway. Normally it is a good thing. So here it is showing you that in normal cells there are two ways to reach to this thing, to keep it alive. So if you attack only A, B will predominate, it will take over that thing. It's like, like two people are doing the same job and one person goes on vacation, the other person will take care of that, and the other person goes on vacation, the first person will take care of that person. But if both the people leave, the work will stop. So if you target either A or either B, cells are alive. But if you target both of them, it is lethal, right? So that is very easy to understand, right? And it makes sense, right? Now, what happens in cancer cells, that normal cells, so here in this case, this is normal cell, gene A and gene B, they are both functional. So if you inhibit only gene A, normal cells will survive. If you hit only gene B, normal cells will survive. Now, here is the thing. Remember I said Achilles heel for cancer. So what in cancer cells, what happens that, so here it is showing you if you make mutation in gene B or gene A, in both cases they are okay, right? But if you kill both the genes, then they will die. So now let's go to cancer cell. So this is normal cell. So normal cell, if you have gene A or gene B only, so if you hit only gene A, then it will survive. If you hit only gene B, it will survive. But if you hit both of them together, they will die. This is normal cell. Now let's come to cancer cells. So gene A, gene B are required for viability. If you hit gene A or gene B, gene A, it will still be viable. But here in tumor cell, gene B is already dead. Okay, they rely on gene A for cellular viability. So now you make drug against gene A. So normal cells will live because they still have gene B, but tumor cells will die because they don't have gene A to start with. Is it making sense? I'm teaching you synthetic lethality in very simple words. So two pathways are present in normal cells. Killing one will keep the other path alive. But tumor already have one pathway. This is gone. So if you hit this pathway, tumor cells will die, normal cells will not die. That is synthetic lethality. Okay, so so let's look at, I will come to synthetic lethality. There is a whole lecture on synthetic lethality. So if you identify what is wrong with cancer cells, so there is a very famous example, and I will tell you about this in details later on, but I want to share just a little bit of more about synthetic lethality. So remember I told you about BRCA1, BRCA2, right? So BRCA1, BRCA2 are absent in breast cancer and ovarian cancer patients, and it is present in normal, like healthy people, right? So what are BRCA1, BRCA2? They are responsible for DNA repair. So when BRCA1 and BRCA2 are absent, then there is another protein called PARP. So PARP takes over BRCA1, BRCA2. So whatever repair BRCA1, BRCA2 does, PARP also does the same, right? So DNA is repaired. So hopefully in all of us here, BRCA1 and BRCA2 are active, PARP is active. So we have two ways of repairing our DNA all the time. Now in Angelina Jolie, PARP was not there, right? It was defective. The only thing here was, BRCA1, BRCA2 were not there. PARP was the only one there. So now the cancer cells are dependent on PARP only for DNA repair. Is it making sense? Now because BRCA1, BRCA2 was not there, they had more DNA damage, so they were more prone to cancer, right? Assuming they developed cancer because DNA repair is inefficient. PARP is there to fix it. Now what happens if you give PARP inhibitor? no DNA repair. So if you give PARP inhibitors to tumors, what will happen? Synthetic lethality. They cannot repair their DNA because you have killed the second pathway also. Normal cells, if you give PARP inhibitors, good. BRCA1 and BRCA2 will take over. So normal cells will survive, but the DNA, the cancer which lacks BRCA1 and BRCA2, they will die because you have killed. That is synthetic lethality. Okay, that's a very simple way of explaining it. Go ahead. Very good question. Yeah, very good question. So what happens is that when you mutate the DNA, right? So let's say you have billions of cells and they are all mutating in different ways. So some mutations will kill the cells because those mutations will make non-functional or this or that. But out of hundreds and hundreds and hundreds and hundreds of different mutations, there will be some mutation which will make the protein more active, more oncogenic. Excuse me. So all other cells will die which had wrong DNA mutation. But the right DNA mutation, which makes the cell more proliferative, will survive, right? So what cancer cells do, they make all sorts of mutations because of DNA damage. And those which die, they die. But the one which survives now becomes almost immortal because now they have turned on something which is not affected by other things. So anytime we give a drug and they go mutation, so they don't know what mutations to make. So they make many different kinds of mutation due to genomic instability. So they are randomly making mutations. Many of them dies, 99% die, but 1% survive. That is a resistant guy, right? So when DNA damage occurs, there is always death around. There's always death around. Tons of cells will die, but then the few survivors will emerge, right? So that is why cancer can survive, because it undergoes mutations. So you are right, not every mutation. If I have to guess, huge percentage of mutations are lethal, but the few survivors, they are the one which makes up a whole new colony of survivors, right? Okay, let's talk about tumor microenvironment. So this is a nice picture of tumor microenvironment. These are blood vessels, and see how many different kinds of cells are there which are making this tumor environment here. Okay, so this tumor microenvironment is made of many different cells, red blood cells, ECMs. This is the extracellular matrix which provides nutrients and all these kind of things. These are different kinds of cells which help tumors. So immune cells come. There is a kind of immune cell called M1. So M1 is supposed to kill the tumor cells. They secrete factors and change M1 to M2. Now they are helping the tumor to survive. So all kinds of things they become, and this is the blood vessel which is feeding that, providing food and everything. Okay, it is like you go and live in a new location where there is no plumbing, no supply of food, and then you make the roads, you do all the things, make it a very conducive environment to survive, and then they spread out. Okay, so now you can actually know what are the players of the tumor microenvironment, and then you can actually develop therapy to target this tumor microenvironment. And so you can also develop drugs which is actually targeting the tumor microenvironment. Okay. Yeah, go ahead.
You are saying tumor microenvironment, we are targeting the tumor microenvironment. But the same microenvironment may be present in other side of body and other parts. So that will be targeted also.
So tumor microenvironment is more unique to the tumor. That is why it is called tumor microenvironment, right? So this kind of environment is not present in other parts of the body. Some of the cells may be there, but that environment is very unique to the tumor. So a person who does not have a tumor, or the body which does not have the tumor, that part, they don't have that same environment. So tumors have a different microenvironment, that is for sure. So the what you are saying, the constituents which form the tumor microenvironment, they may be present in other parts of the body too, and they are definitely present. But if you target this one, they are affected more than those guys in different places there. Is it making sense? Yeah, so some of them are there. That is why most of this therapy have some side effect. But then you have to kill the greater evil for that less side effect. Okay. So that is tumor microenvironment. And so see angiogenesis, killing of blood vessels. This angiogenesis is a normal process in rest of the body. So when you give angiogenesis inhibitors, of course the rest of the body will be affected, but it is not that dependent on angiogenesis as the tumor. So people still survive, right? I am in touch with a patient who is undergoing immunotherapy and angiogenesis inhibitor therapy right now. And I talk to that person pretty regularly, and he's feeling better day by day. So just killing the cancer, side effect is less compared to the cancer-related pain and everything else. Okay. And finally, the epigenetic modulators. So I have already told you about epigenetics. So you can actually give inhibitors to control epigenetic modifications. Okay, so this is like, so once again, epigenetic could be DNA methylation, histone modification, chromatin remodeling. I'll tell you about chromatin remodeling a little bit later. So you can actually give inhibitors. So you can give methylation inhibitors, okay? Or you can control acetylation by giving those kind of inhibitors. So now I'll tell you about the Achilles heel concept. So what that means is that cancer cells, in order to survive, they have new mechanisms which help them survive under adverse conditions. But when they develop the new mechanisms, they also develop new vulnerabilities, right? So if you can identify this new vulnerability, that is the Achilles heel. So a person is very strong, okay? This is a Greek, I think it is a Greek mythology. So this person is very strong, but the only weakness he has in this heel here. And if you can identify that vulnerability of cancer cells, you attack exactly at that weakness, and then you can kill the cancer cells. Okay, so for example, this brings us to synthetic lethality. If you target A or B, normal cells will survive, but cancer cells only depend on one target. And if you target that one thing, because they don't have the support of both targets, if you target that one, then cancer cells will die. That is the Achilles heel here. This is another very good example. So cancer cells, right? Because in order to proliferate rapidly, they have huge amount of polyamines in that, because polyamines are used for constructing the whole cell, right? And normal cells have low polyamine contents, so just simple polyamines, right? And these are used as the building blocks to make other macromolecules. So now we know cancer cells have high amount of polyethylene. So there is this BSO enzyme, BSAO. What it does, it takes the amines and it changes it to aldehyde. And if you make too much aldehyde, what does aldehyde do? Aldehyde is very toxic, and it cross-links a lot of things, react. Aldehyde group is very reactive, right? It takes an amine group and it will form Schiff base with that, right? So you treat it with BSAO. Normal cells don't have too much amine, they will not form aldehyde. But cancer cells have too much amine, they will form too much aldehyde, cells will die. So now you are targeting the vulnerability in cancer cells, right? So if you identify the weakness, so they need for survival huge amount of amines, and they make it. Now we attack those things, right? We have identified the modification there. Okay, so that is what. So what are the emerging frontiers there? We keep on monitoring the patient regularly. We take regular biopsy to see how they are responding, what mutations, what things, and we accordingly keep on changing the drug treatment. You adjust the dose. You see what time, if you start the drug, how soon it is responding. Should we add something? Should we remove something? So all of these things. AI can So, I was working on something, and then somebody told me that, yeah, this is very effective, but this is very sad story that they are going to develop resistance towards your therapy too. So if you can proactively think about that, if I inhibit this, what kind of resistance mechanism can emerge, then you can proactively design that route. And AI can do easily, and actually you can do without AI in the lab also. And I can tell you the ways to do it. So that will very help. And then you can combine many different things, even in immunotherapy, to do that. So resistance is inevitable, that we know, but we can definitely beat it. No doubt about it. Okay. And the Achilles heel lies in the new weaknesses of cancer cells. They think they're very smart, but they also have new weaknesses. And if we can identify, we can attack that. Okay. And that will result in long lasting that will extend our life, and we'll have better quality of life, and we can beat it in the end. All right, we are leaving it in a better note right now for lunch. All right, guys, so we can have lunch, and then what is our schedule again? 2 to 3 and 3:30 to 4:30 or 5:30 or 5, whatever, we'll see. Yeah, go ahead. You speak very softly, you even beat me.
I was trying to compare the bacterial resistance with
Yeah,
cancer resistance.
yeah.
In the case of microorganisms, one is bacteria, one resistant to
Drug.
that one is spread because it is contagious, it's infectious. So later we end up synthesizing a new generation of drugs because the target is uniform. But in the case of cancer, if mutation happens in one person and resistance occurs in another, how do we end up having synthesizing another generation of drug, yet it is not contagious across cancer?
Very good, very good question. So I don't know who asked me. I'm sorry, I talked to so many students, but somebody said here that Gleevec introduces 5 mutation. Who said that? You said that. So remember I was telling you about Gleevec story, and I said one mutation develops. And why I said one mutation? Because I was very close to that mutation, because in kinase field it is the residue which changed, that amino acid which changed is a very famous residue called gatekeeper residue. And we developed a method to target the gatekeeper residue. So when that mutation occurred because of Gleevec treatment, I had a very nice discussion with my advisor about that thing. So we are very connected to that gatekeeper mutation. But in reality, what happened, and that will answer your question. So when Gleevec was given to patients, many different kinds of mutation appeared across the patient, this thing. So one mutation here, in some patient this was the mutation, in some patient this was the mutation, in some this was the mutation. And remember, there are 20 amino acids, so all combination, even if the residue was the same, the mutations were very different. So what happens is that Gleevec binds like this. So there are 5 mutations, let's say, emerged. So now you are making the next generation compound. So it is possible that this next generation compound will not target all the 5, but it may target 1, or in the best case scenario 2, right? But then you make some other which will target this, some other compound which will target this. So it's a constant fight. You make several compounds against whatever mutations you identify. Then you do the genetic profiling, find which mutation, you change the drug. Okay, but there are few hot spots. The whole protein will not change. Okay, the whole protein will not change. There are few spots which are more vulnerable to changes, and likelihood is that these spots are going to mutate more. So if you can target those, depending on which mutations are identified, you have, your tool set has more tools in it, and you can accordingly use it. Okay, so you are right, cancer is not that simple that you can cross like antibiotics, right? But yeah, it can still be targeted. And now with this, all this AI driven thing, and the screening and the computational tools are becoming so powerful, that you can actually design molecules which may be very effective, and that answers your question. That if you combine the two things, you can proactively design. I know people who do virtual screening and they make their living out of virtual screening because it can be that effective. And people do virtual screening, and out of millions of compounds, they identify the compounds. But these are virtual screening of real existing compounds, right? And so you can actually procure that compound and it really works. So because now we have, our library is increasing tremendously, right? Our computational tools. Now computational tools, they feed from the real data. So the more real data you create, the more sophisticated these tools become, right? Because now they can predict protein flexibility and everything. So you make more and more compounds, and then you can change it. But keep in mind, this is human life we are talking about. So even if we can save some out of others, we are saving lives, and we are saving the lives of their families and loved ones, right? What is the common thing about bacteria and cancer? They mutate very fast. And like we said, even if 99% die because of the mutation, that 1% is more than enough. That 0.01% is more than enough to build a new cancer. And bacteria, oh my God, they divide every 10 minutes. So even if millions of them die, trillions will emerge in no time. So, all right, let's stop. Otherwise, we'll cut down the lunch time. Okay? If you can take a cold tumor and you can make them a hot tumor, then more targets will be exposed and you have more chance of treating that one. So that's what this means here. And sometimes epigenetic drugs, that means the drugs which modify the epigenetics, can be useful. So epigenomics means you are looking at, whenever there is this omics word, that means you are looking at it globally in the whole organism or in the whole cell. So this is a cartoon here which show that this man is a happy mind. This child is a happy child initially, and then early life crisis, some kind of abuse, whatever abuse it is, that created huge adverse condition. And those adverse condition led to CPG methylation, so epigenetic changes in that body. Okay, so the DNA sequence is still the same. You can imagine the twins were born. This one child was raised in very happy environment, right? And he became a very happy going person, very successful. This child, same intelligence, identical twins, but he faced a lot of adversity, childhood trauma, things like that. So his brain chemistry changed in terms of epigenetics. DNA is exactly the same, but epigenetic modifications are different. So when this person induces is, so we all face stressful conditions, okay? I know of a person, the more stressful condition this person faces, the more happy she becomes. So her boss is very abusive, and she tells me a story. And when she's telling me the story, she's laughing. And then she's laughing more, and then she's laughing more. So she said, Didi, this happened. अरे भैया तो मुझको पहले बता देते, मैं तैयारी कर लेती, क्यों मुझको 1 मिनट पहले बताया? So every stressful condition is a joke. So she knows how to deal with stress. She's never stressed. And I always tell myself, if I face the same situation, I'll be super stressed. Imagine you're asked to teach a class 5 minutes before the class starts, and you have no idea what to teach, and they throw you in the meeting without telling, oh, speak in front of 100 people on this topic, and you don't know anything about that topic, and that person knew about that topic 10 days ago. but he's deliberately not telling you to put you in that stressful situation. We all will be stressed. This woman not stressed. She said he needed to give me 20 minutes. I would have prepared, right? And she starts laughing and laughing. And now what happened? After a year of this kind of abuse, now this person is actually a friend because he knows nothing will perturb that woman, and she doesn't have any enmity towards this person also because she's genuinely a happy person. But this person who has faced a lot of trauma, he has so many stress receptors in his brain that small stress is amplified in his brain, and eventually that person cannot handle that. So this is due to epigenetic changes. It could be due to epigenetic changes in the brain. So that is why we all react differently to stress situations. Okay, so epigenomics is tightly regulated with cancer. Many people believe that if you make epigenetic drugs, you can really change cancer. Okay, and epigenomics, okay, that promotes cancer in many different ways. Epigenomics changes can lead to uncontrolled proliferation. It can lead to uncontrolled metastasis. It can prevent cell death. It can change immunity. This is true. When we are stressed, we are more susceptible to diseases that you may all have seen. When I'm stressed, I get cold, I get this, I get that, right? Oftentimes we have fever because we cannot deal with stress. So our immune system goes down. So it evades immunity, it induces angiogenesis, so it promotes cancer in many different ways. Okay, so this is the part of epigenomics. This contributes to cancer. Okay, so epigenomics. So you all know about different kind of modification. So I barely told you about DNA methylation and histone modification, but there are other epigenetic mechanisms also that contribute hugely to diseases. And these are nucleosome positioning and non-coding RNAs. Okay, these days these non-coding RNAs, they are becoming really major players in human physiology and pathology. Okay. Okay, so so epigenomics is also called as the software of the genome. So what is the hardware? The DNA sequence. That's the hardware, and the software, which is the epigenomics, is the one which controls how DNA expresses RNA. Hey, no cell phone. Okay, in my class. Okay, so the epigenomics control which genes will be expressed. So DNA sequence is identical, but that is controlling which proteins are going to be made. Okay, so just to remind you once again, so this is here that many different factors, okay, many different factors control epigenomics. So you may have heard that when women are pregnant, they said in some culture it is very important, especially in Native American culture. So you guys know who are Native Americans, right? You all know Native American culture. So Native American people are spiritually very advanced people, okay? So in their culture, it is essential that the pregnant women do not face any stress. They go out of the way to keep pregnant women happy and stress-free, because if the mother is stressed, it affects the child in the, at that stage. So that is what it is showing during pregnancy itself. Those stress things can be absorbed by the child, and that can lead to lifelong problems in future. Okay, new childhood trauma is a major. I was reading it today in news that Bob Reiner was killed, right? The famous director of Hollywood was killed by his own child. He and his wife were killed. So he actually made a movie on this thing, and he once in an interview famously said, he was a very nice man. So it's very unfortunate that he was killed by his child, and the child has drug problem. So he did his best to help him out. But he very famously once said that all the bad people, like who are bad people, who kill other people, or who are violent people, right? They have all suffered some kind of trauma in their childhood. If somebody has a very happy childhood, they are not likely to develop into a violent person. So it is very important that, the children have normal childhood, and we cannot control it because it is your life circumstances. Many times parents cannot control the circumstances also, despite their best efforts. But childhood trauma can have long lasting effects. We already talked about all these drugs and aging, physical activity, exercise really helps you, right? And once cancer starts, then it is due to genomic problems. It could be epigenomic, it could be environmental thing which affects genetics and epigenetics. Okay, so this you already know. This, see here you have more. uh, See, look at this thing here. So in this small area you have 1400 nanometer mitotic chromosome. Okay, now we open it up a little bit. So now this is the ordered structure. Now we open this structure, it looks like this. We open it further. This is called the 30 nanometer fiber. Okay, we open it more. Now it is beads on a string kind of structure. Now when remove all the histone octamers, now this is DNA, and now you see it there. So this is extremely ordered structure. Okay, so let's focus on this part here. So this is how it looks like. Some part is very tightly closed, some part is more open. So this is more likely to make RNA than this part here. When it is methylated by histone methyl transferases, so remember this is the protein part. So when it is methylated, it is closed, it will not make RNA. Once it is acetylated, now it will make RNA. Okay, so DNA methylation, histone modification, chromatin remodeling, and non-coding RNAs, they control, these are the major mechanisms of epigenetics. Okay, so I already told you about histone modification and methylation. This opens up, this closes it. Okay, so and this is the modification. So this is cytosine is modified, and once it is, this is histone, this is DNA, this is histone, protein modification, both methylation. DNA methylation also closes, histone methylation also closes. Okay, this is cytosine, and then this is the promoter region. So once it is methylated, it is closed. Now RNA polymerase cannot bind, and there is no transcription. Okay, so now you can imagine there, if there is a tumor suppressor, then this is going to get methylated. So tumor suppressor, hypermethylation. Now it is not going to be expressed. If this is not methylated, it is going to be highly expressed. So oncogenes, it will be hypomethylated. Okay, so look at these things here. By looking at this pattern, you can tell which one is a tumor suppressor, which is a tumor promoter. So APC is hypermethylated. So according to you, it is a tumor suppressor or a tumor promoter? Raise your hand if you think it is a tumor suppressor. It is a tumor suppressor, right? Because it is hypermethylated. KRAS, oncogene, right? So you can see cancer cells know exactly which one to hypermethylate and which one to hypomethylate. c-MYC is such a big oncogene, right? Hypomethylated. And also microRNAs. How many of you know about microRNAs? Some of you know about microRNAs, right? So microRNAs are the one which control which RNA will be expressing the protein or not. So what they do, microRNAs can bind the RNA directly, or they can bind some part of the DNA and influence the transcription or sometimes even translation. Okay, so microRNAs do not make any protein, so they are called non-coding RNA. Any RNA which makes protein is a coding RNA, because protein expression of protein is also called coding. So any RNA which does not make protein is called a non-coding RNA. And microRNA influences gene expression in many different ways. So that is why they are not changing the DNA sequence, but it is influencing that. So it is considered an epigenetic mechanism. Okay, so this is DNA methylation. If it is unmethylated, gene is on. If it is methylated, it is off. And, and because of that, they can control oncogene expression, they can suppress tumor suppressors, and they can lead to plasticity and therapy resistance. So epigenetic changes can occur which can make a cancer resistant to therapy. I will ask you now, give me one mechanism by which epigenetics can lead to therapy resistant. Can anyone tell me? Again ask the question. So tell me one mechanism, one epigenetic mechanism by which a tumor becomes resistant to therapy. So it was responding to therapy before, and then some epigenetic mechanism occurred and it became resistant to therapy. So tell me, make up any one mechanism. All of you have enough background, just give me any one mechanism. The important thing is that no matter what mechanism you make up, right, it exists in nature. Because nature is so diverse that you can be 100% sure that that mechanism exists somewhere, even if you think you are making it up. So I will guide you. So remember methylation, DNA methylation, off switch, histone methylation, off switch, histone acetylation, on switch, right? So keep these three things in mind to make your life simple, and then tell me that there is a tumor which is responsive to stress, responsive to therapy, and then it stops responding to that therapy. So I can say one non-epigenetic thing, or that protein mutated, it no longer, the drug that you were giving no longer binds there, and so it is resistant. So I just gave you one mechanism which is due to mutation, but it is not epigenetic mechanism, right? Because epigenetic has nothing to do with changing the DNA sequence. So I ask you, give me one epigenetic mechanism. Any one of you. So I want you to think in backward. You know retrosynthesis in organic chemistry? So what is retrosynthesis? You start with the molecule and then you break it apart and then you build the molecule based on that. So I want to do a retrosynthesis like of this thing. So suppose you have a tumor is responding to something. So what is that something? Let's go retro now. Maybe it is due to a protein, right? Yeah, ligand maybe or something, right? Now think backward. So it has become resistant to that therapy. Which way? We cannot use DNA mutation because it is an epigenetic mechanism. Yes, so now this receptor is no longer even there, and you can keep on adding drug, and it has expressed some other protein, it has rewired itself. That is the most important mechanism. Or you can do one thing also, that you know, it has in normal cells, this efflux protein, their promoter is methylated, so they are not expressed. Now they have removed the methylation from those, efflux pumps are expressed and the drug is pumped out, right? So that would be an epigenetic mechanism also. So you can think of many mechanisms by which it can lead to therapy resistance, right? And plasticity is, again, I will give your example. Like they don't even make the protein anymore. The receptor is absent and now they are waiting on something else. They make some other protein and that becomes the driver for cancer. That is plasticity, right? So you already know how to do that. So here this is normal transcription, TSG is expressed. TSG is tumor suppressor protein. And here you have methylated, tumor suppressor protein is no longer expressed, right? Here in normal cells, oncogene is not expressed because this is hypermethylated. They remove the methylation in cancer cells, now oncogene is expressed. So this is what it is showing here. So promoter methylation is one of the major ways they control gene expression. So here it is methylated, tumor suppressor is not expressed. Okay. Now let's talk about other mechanisms. Acetylation opens it up, more transcription. This I have already told you. So if you have to block this thing, somebody was asking me, how do you block DNMTs, the methyltransferases? So here, if you block this methyltransferase, then this is not going to be methylated, and if it is not methylated, tumor suppressor will express, right? Here you have to do the reverse there, okay? So these two you know, DNA methylation, and what is chromatin remodeling? So I think I have included one figure here. Let me see. So look at this structure here. So this is heavily modified. Now once this is really tight structure. Now once you add the acetyl group here, so sorry, this round thing, this is methylation, and this pentagonal thing, this is acetylation. So when it is acetylated, it is open. When it is methylated, it is completely closed. This is what it is showing. Now look at the structure here, okay? So histone is modified. So I kept epigenetics very simple, but actually there are many more modifications. There are at least 20 different kinds of modification which happens on histone. They can get phosphorylated, they can get methylated, they can get acetylated, they can get sumolated. Sumo group is a different kind of this thing. They can get ubiquitinated. You know, ubiquitination is because of protein degradation, but there are different kinds of ubiquitinal groups, and some of them turn on something, and they are also signaling, which has nothing to do with degradation. So look at all different kinds of modification: acetylation, methylation, phosphorylation, ubiquitination, isomerization, ADP ribosylation, deamination, S-methyl cytosine, and SOMO is not even written here. So there are many kinds of modification, and that is called as histone code. So when there are many modification, it's not as simple as on-off switch, but it is a whole pattern of things which determined like how gene expression is controlled. Now look at the microRNA. So this microRNA, so suppose this is the promoter region from your view, this is the promoter region, this is the gene. So a microRNA may bind to the promoter region and inhibit transcription, right? Or the microRNA can bind to the RNA which is formed and it can degrade it. Or it can sometimes activate also. So microRNAs are also controlling gene expression. Okay. So this you already know, acetylation can lead to oncogene activation. So what about HDACs? So HDACs are histone deacetylases. Somebody asked me yesterday, I don't know who asked me, you asked me, right? So how do you decide? So I am telling you a very simple story that acetylation increases transcription, methylation decreases transcription, right? But things are not that black and white in reality. Sometimes acetylation can also occur in tumor suppressors, right? So tumor suppressors are also expressed more. So it is not that histone acetylation specifically only happens for oncogenes. It can also occur on tumor suppressors, okay? So in that case, the reverse of histone acetylation is histone deacetylases. So histone acetylase, they add acetyl group, this enzyme removes acetyl group. So suppose histone acetylase histone these are called HATs. HATs are adding acetyl group on those proteins like tumor suppressors, and they are expressed. So that's very good. So if you add HDAC inhibitor, then if they remove acetyl group, tumor suppressors are not going to be expressed. So what I'm trying to say is that it is not black and white all the time, that acetylation only occurs for oncogenes. Sometimes it can also occur for tumor suppressors, which is good, right? But it is also occurring to some oncogenes. So how do you decide? The question you asked me yesterday, how do you decide if acetylation is doing both oncogene and tumor suppressor? If you inhibit that, is it good enough? In that case, you have to give the drug and see which one is more effective. like killing the oncogene and killing the tumor suppressor at the same time, because you have no choice because it is targeting both. So in that case, you have to actually determine experimentally, and it may have some side effect, but in reality, it is not crystal clear in every case. There's always some kind of good thing also going on. So that is why it is asking, is it a friend or a foe? Okay, so HDAC can also lead to cell death. It can promote angiogenesis. So there are many things which are controlled by histone deacetylases. We are not going to discuss that. So it is always, you have to weigh good versus evil when you are deciding a therapy. That's the point here. Okay. So now the thing which I did not tell you about was chromatin remodeling complexes. So what happens is that when I showed you this structure here, so in reality DNA is like this. It is like this, right? It is very, very, very complex, and it is extremely folded. Okay, so when you unravel the DNA, right, sometimes if you put something there very far away from the transcription site, that perturbation can have effect here also. So that is what is shown here in chromatin remodeling complex. In this one here, so see, there are many proteins which bind the histones. So look at some of these conditions here. So here it is trying to see, these are histone octamers, right? So there are these proteins which will take out the whole histone out here. So this, they have removed the whole this thing. So obviously it is wrapped around this, and if you remove the whole protein, now there is nothing to be wrapped around. The whole structure will fall, right? So these proteins are called chromatin remodeling complexes. They will come and they will just take the histone away from that, and that it will open. I can see you guys are very tired. We all need a cup of tea to keep ourselves awake. But let's,
3 o'clock.
okay, 3 o'clock. We have to survive till 3, then we get tea. Okay? So, okay. So, so there are several proteins which will bind chromatin structure, which will distort it in this way, that, open it up, right? All kinds of drama is happening which can change expression of gene, which can change gene expression. Okay, so let's briefly talk about these non-coding RNAs. So Pol2 is RNA polymerase, right? The reason it is called Pol2, because in humans, do you know that there are 3 kinds of RNA polymerases, 1, 2, and 3? And RNA polymerase 2 is the one which makes messenger RNA, and messenger RNAs are those RNAs which make proteins. Okay, so they are called the coding RNA. Okay, so once messenger RNA is made, okay, I don't know why it is showing long non-coding RNA. I'm 100% sure RNA polymerase 2 is the one which makes messenger RNA. Do you guys remember which RNA polymerase makes messenger RNA? Second one, right? So non-coding RNAs are, I think, not made by, or maybe they are also made by RNA polymerase 2, because yeah, along with mRNAs. But this ribosomal RNAs, they are made by partly by 1 and 3. 5S is made by 1, and then other 3 are made by other. So yeah, so this RNA polymerase 2 also makes some non-long coding RNAs. So these are the RNAs which, see, look at this thing. This is mRNA, right? So human mRNAs, eukaryotic mRNAs have a cap at the 5' end, and they have a long poly A tail. You remember? So this is a human messenger RNA. So this is binding this one, and because of this binding, it can stabilize it depending on where it is binding, or it can degrade it, or it can change the expression of this gene. So this guy has nothing to do with this mRNA. But once the mRNA is formed, it can control what happens to this mRNA, right? So this is the long coding RNA, and this can also degrade this one here, okay? And then this RNA here, this is bound to this one, and then this is bound to another protein. Because this RNA is bound, now a protein binds and it is activating transcription. Okay, so this is binding to histone directly, and so you make an RNA which then opens up the chromatin, and then it promotes transcription. Here it is the other way around. It binds, now it calls a repressor, closes the chromatin. So basically non-coding RNA, we are not changing the DNA, but because of this long non-coding RNA, we are controlling transcription, right? So now let's compare genetic mutation with epigenetic changes. Okay, so in this one, genetic mutation is a permanent change in the DNA because you have mutated the DNA, correct? And this can caused by errors in DNA replication or exposure to radiation chemicals, or one can inherit from the parents. Okay, and this is permanent, it can be inherited, and because you have changed the genetic code, it will change the protein formation. So for BRCA gene, they are mutated in breast cancer, they don't work. Do you guys have you guys heard of sickle cell anemia? So sickle cell anemia has mutation in hemoglobin gene, and because of that they are sickle shaped, right? So normal red blood cells, do you know how long of a half-life time they have? 120 days, very good. And what is the half-life of sickle cell? 10 days, roughly 10 days, right? You guys tell me, how many days? Anyone remembers? I also remember with confidence 120 days for healthy ones. How many for sickle cell? As I recall, it is 10 to 20 to 30 days. Yeah. So just one mutation and you change it, right? But if you compare it to epigenetic, like DNA modification, non-coding RNA, this is reversible. Okay, this is reversible thing. This is not inherited, right? Sometimes it can be inherited, but most of the time it is not inheritable thing. Okay, and example is hypermethylation, hypomethylation kind of thing. So this is comparing both of the things. DNA is changed, DNA is not changed, right? This is permanent, this is mostly reversible. Okay, this is, it can be inherited or DNA repair mechanism. This is, look at the epigenetics, environmental, diet, stress, drugs. Okay, so I work with a kinase called CDK5, and CDK5 kinase is very good kinase. So when you guys are studying for exam, CDK5 activity increases quite a bit, and that is why your memory increases very high, right? So it's a very good kinase, but if it is always in the high state, active state, the brain cannot handle that much activity and the brain will degenerate and the person will get Alzheimer's. So CDK5 hyperactivation can lead to Alzheimer's. So this activity is ideal. If you have less CDK5 activity, then what will happen? You will have disease. No, neurodevelopmental of this thing. So one person can have schizophrenia. They can have, all these kind of weird diseases, ADHD kind of things. So these kind of diseases can develop if the activity is less. This is normal. And this much activity for a long time, when we have exams, it is slightly elevated. Exam stress is gone, it comes back. So that we can handle, right? But if it goes high all the time, that's when the brain will degenerate. Okay, so but CDK5 people who take drugs, okay, CDK5 kinase is the one which makes the pathways which causes addiction. So CDK5 is responsible for a person get addicted to drugs. So drugs can change the whole neuronal pathways in the brain, and then they will crave for more and more and more drugs. Okay, so so many times that leads to epigenetic changes where the person is really craving for that drug. Okay, so this, in mutation, you will get a different protein because you have mutated the gene. But here it controls which gene is going to be expressed and which gene is not going to be expressed. It's not changing the protein itself, but it is controlling the expression level of that protein. Okay. So just see BRCA1 mutation in cancer, BRCA1 promoter methylation in cancer. This is a very good example. You methylate it, BRCA1 is not expressed. BRCA1 is there, healthy, no problem. But you hypermethylate it, it's not going to be expressed. Okay. So let's look at the consequences of epigenetic changes in cancer. We already know silencing of tumor suppressors. We have seen activation of oncogenes. Okay. So go ahead.
To identify how you will identify that if someone gets breast cancer, if the gene get mutated, we will find that by unique technique. But at least what technique we will use for that if we are genetic change.
That is very good question. Okay, so I will even show that thing there. That is very good question. So how will you know it is epigenetically modified? So we have actual techniques which will tell you whether the promoter region is methylated or not methylated. So what you can do, I will tell you very simply, right? So I will tell you about those techniques, I think in this slide, in this presentation only. But in addition to that, what you do, if you do genetic testing and you say BRCA1 gene is there and you can say it is not amplified and you can see that it is not mutated, it will give you all the information. But then you do proteomics and you see in normal things it is expressed and it is not expressed at all. Or it is expressed at very low level. But you see the gene is intact. So now you can guess that something is blocking its expression. So you can guess it is probably methylation, right? And then you can figure it out by actually looking at the methylation pattern. And there are ways to do that. And you can look at all the methylation across the genome also. Okay, so these techniques exist. Okay, so you already know this one, so I'll skip. You already know the activation of oncogenes, so I'll skip. Now, I did not tell you about genomic instability. So whenever, what is genomic instability? So what happens is that genomic instability is that in general terms, the cancer cells are very, they're always changing themselves, mutating, in order to become survival of the fittest. But when they do that, obviously some mutations are lethal and they get eliminated. And they don't care about elimination because even if 1 or 2% cells survive, they have acquired those mutations which really enable more and more proliferation and more resistance to adverse conditions, right? So in terms of epigenetics, so why you can gain genomic instability? So when the DNA is replicated, it is randomly modified epigenetically, right? So it can lead to double strand breaks, chromosomal rearrangement, cancer. So a lot of things can happen just during normal process because of the change in histone modification. So the whole genome will be unstabilized, okay? So some of them will die because of that. But in other cases, they will acquire resistance and they will become even more aggressive. So epigenetic changes can change the whole genomic structures, okay? So because of that change, so for example, this is a tumor cell here, okay? And this is, remember yesterday I said that normal cells are pretty differentiated. So if you look at mesenchymal cell, they look like this, right? The more cancerous they become more round. They don't look like the original parental cell. They look like more round, more aggressive kind of thing, okay? So this is a differentiated tumor cell, and then now we gave different kind of radiation. Most of the cells died, and now this stem cell emerged. Remember, 99.99% cells die, but 1 or 2 cells survive, and they are resistant to all therapy, all radiation. These are the stem cells. And because they are stem cells, they now will form new kind of, they can modify themselves into different kinds of cells. In some cases, they will have this, these cells are different from these kind of things. So these stem cells, they have infinite renewal property. So they can keep on making different kinds of stem cells. And then they are also resistant to therapy. So these stem cells are very aggressive. Okay, they make some cells which are extremely aggressive. They will form metastasis. Here they have changed even their phenotype. Remember, androgen receptors have disappeared, something else has happened. So they have changed their phenotype. Now you give this kind of stem cell, see, they are all originating from the same parental cell. How many different kinds of things they have done to themselves. In this case, this is not even forming tumor, and this is forming tumor, but look at that, so many different kind of heterogeneous tumor, right? Here you have given chemotherapy and radiation therapy, most of the cells died, only one survived. But then this is again going to make different kinds of cells. So when you have different population, if you kill one, the others survive. If you kill the others survive. If you kill the other, all others survive, right? So that gives them infinite quality. So these are the properties of these cancer stem cells. Cancer stem cells the hardest to treat, okay, because they can acquire different things. They can renew themselves. They are resistant to almost all sorts of therapies, okay. Now immune evasion can also occur due to epigenetic modifications. So let me give you an example, or let me ask you an example. Give me one example by which epigenetics can lead to immune evasion. So let me remind you, let me give you a clue, and then you connect the dots, right? So yesterday I told you about PD-1 and PD-L1. Okay, so let me remind you about PD-1 and PD-L1. So T cells are the fighter cells. T cells are the immune cells, right? So their job is that anytime there's infection, they will go there, they will fight it off, right? So they get so charged up when there is infection that they are supercharged. So initially it is great to be supercharged because they go to the side, they defend us and everything, but they are still too much momentum going on. So if they're on for very long, if you're working in very high speed, it is, right? So it will lead to autoimmune disease because they will start attacking that because they are in that fighter mode. So that's when APC cells come and they quieten the T cells. So they have these receptors called PD-1 receptors. So if you don't touch the PD-1 receptors, they are in the hyper mode if I am a T cell. Okay, so these AP1 cells will come and they have a ligand for PD-1 which is called PD-L1. So if they come and bind there, I will immediately become quiet. So tumor cells, what they do, they're not supposed to have PD-L1, but they will express PD-L1. So anytime T cells go close to them to kill them, they lock horns with them. And T cell thinks, oh, I'm supposed to be quiet, and they become quiet, and there is no tumor killing. So now apply an epigenetic mechanism. How can you use this using epigenetic mechanism? Anyone? Exactly, and now it will be overexpressed, right? Great. Give me one more mechanism, again for PD-L1. What else can happen? Hypomethylation of the promoter region of PD-L1, right? So you guys are all with me. So you can think of many epigenetic mechanisms just based on whatever we have studied so far. So it is not surprising that immune evasion can occur due to epigenetics. What is epigenetics? Oh, you have perfect DNA, but I have the control. I will decide which protein is going to be expressed and which is not going to be expressed, right? So DNA is there, but I am the decider of the DNA, right? So that is what it is shown here, that there are many different mechanisms, okay? So that is what is craved from T cell thing, right? PD-L1, PD-1, okay? That can also lead to resistance to immunotherapy through PD-1, and I told you briefly about CTLA-4 yesterday, okay? So this one is, so you know what happens? Our immune system is surveying our whole body at all times. And anytime it says anything which is out of place, it will recognize it and it will eliminate it, and we are safe. When the enemy is very strong, it will fight, it will fight. Sometimes we'll get fever also, we'll feel down for days. That is immune system fighting off the infection, right? When you get cold or something, you are so drained, you don't have energy, your immune system is fighting the virus of cold, right? So that is why it takes too much strength, right? And our body is inflamed and all these things. Everything comes with a cost. When a lot of fighting is going on in the body, our body is drained. When you get a vaccine, we all get fever for a day or two, right? Most of the people prefer to have vaccine on a Friday so that if the fever comes, they have the weekend to sleep it off, right? So, and we have to plan our vaccine too, that we assume that we'll get fever. So we decide accordingly towards the end of the week so that we can at least get rest in case that happens. Okay, so but then the cancer cells are pretty smart. So even after the fight, they quieten the T cells and the tumor grows. Okay, and then it metastasizes. And then you can also plan the drugs. But then even with PD-L1 drug, right? It is such a good drug. But eventually cancer cells will develop resistance towards immunotherapy also. Okay. If you are attacking PD-L1, PD-1, they will express CTL4, something else, to evade that thing. Okay, so that was immune evasion. Okay, now this is showing you some more immune. Do you know what is innate immune evasion versus? Can anyone tell me that? Innate and the other one. Tell me what is innate. Intrinsic immunity, very good. So suppose we are attacked with something, our body, heaven forbid, if we hurt ourselves, right? Immediately macrophages come in and they do a lot of things. Blood clotting happens and macrophages come up, they eat all the garbage. That's innate immunity. And what is the other one? Acquired immunity. And what is the meaning of acquired immunity? And then what happens? Yes, that is the main thing. Acquired immunity is not immediate, so that cannot come into play. Depending on what we are infected with, then our immune system will digest that foreign material. Then the antibodies will be made against those foreign thing. Then the whole thing will be cleaned up. And then the beauty is that they will save those B cells, the antibody producing cells, for future also. So we will always have the memory in this lifetime against that disease. So that is why when we were vaccinated, when we were 10 days old, we are still resistant to that because we still have the memory B cells there. So we will never get that disease unless the virus changes its thing. Okay, so that is what it is shown. Okay, here. So T cell trafficking. So you can actually so this is evasion. Cancer cells antigen release, cancer cell antigen presentation, priming and activation. These are the things. Okay, these are the things. So you can actually develop therapy against cancer cells. So cancer cells sometimes release specific proteins which are specific to cancer cells. If you can recognize that, you can develop a vaccine against it, or you can develop T cells against that antigens also. But then what will happen when you give the therapy, cancer cells will stop expressing those proteins. So now they will evade that also. So it's a constant fight. Okay. So then look at these epithelial cells. So they are changing their phenotype. Now they are becoming mesenchymal cells. This is EMT, epithelial to mesenchymal transition. So if you give drugs for a very long time, now they have changed their thing. They are very motile. They can squeeze through blood vessel. They can squeeze through small holes. They will go somewhere else. They say, okay, this room is very hostile now. Let me change my thing. Let me sneak through the door. Let me sneak through this window, become very thin, very flexible, go out and make my colony somewhere else. And then move from there and make somewhere else. That is metastasis. Okay, they find a new environment and they just colonize there. Okay, and so these are different proteins which are involved in that. Don't worry about this thing. But the more mesenchymal they are, the more aggressive they are, and the more motile they are. And more motile, more invasion, more metastasis. Okay, so it is showing that you have EMT signals, and see some of the cells, they escape, they come in the blood vessels, they come out of the blood vessel, and they form a tumor at some other place. So when someone has liver cancer, they metastasize in the lungs first. When someone has breast cancer, those metastasize in the bones. Okay, so different kinds of cancer, they metastasize at different places. Okay, one time a student of mine, she showed me, so I'm not a doctor, right? So I'm not in touch with cancer patients, which is good for me because I don't, it's very painful to hear that, right? But in that case, so she had a relative who had cancer, and it has metastasized. So I know all the principles. So she was from a different country. So she asked that person to send the PET imaging scan. And I saw that scan for the first time with my eyes, the actual data, right? Not from a textbook, but actual data of a person. And the cancer had metastasized in all over the body, like here and here and here and here and here. And that was very painful to see. You really pray that you don't have to ever see that thing. I mean, unless you are a doctor. But it was like, it can spread all over the body. And sometimes what happens is that we have so many drugs that we prolong the life, but it is not worth prolonging when it has spread so much. I mean, we can say that, but for family members, every day counts. You really want to prolong the life of your loved ones. Okay. And so this you already know. You have already given me ways by which it can lead to therapy resistance. Okay, so that was the therapy resistance that can actually occur in many different ways. All right. Now I answer your question. How do you know, right? So chip sequencing. So there are DNA methylation arrays and bisulfite sequencing, which will tell you DNA methylation exactly. ChIP-6 you have probably heard. It will tell you about histone modification. ATAC sequencing will tell you about chromosome accessibility. Please send those things to the students today, and I will send you today, provided I have access to this thing. And you can even do single cell epigenomics. This is the last slide, I think. Okay, functional, this thing, we'll talk about this. Okay, just I think 2 more minutes. So DNA methylase inhibitors are already in clinical trial, and some of them are approved. They are histone deacetylase methylatase, and there are combination therapy of epigenetic drugs with immunotherapy or targeted therapy. And they are BET inhibitors. So what are BET? This is very important. So bromodomains are the ones which bind acetylated histone. So if you give BET inhibitors, these acetyl groups are exposed and they can be removed also. Okay, so BET inhibitors. So BET proteins are epigenetic regulators that bind to acetylated lysines. Okay, so if you remove the BET group sometimes, the acetyl group is too exposed and they can be deacetylated. And if they are deacetylated, the chromatin will close. Okay, so that way you can prevent activation of oncogenes. Okay, I will stop now. So this is, you guys are going to get this presentation. So if you have any questions, I can answer it later. Okay, all right, guys. So it is tea time. All right, let's have some tea, and then you guys can ask me more questions. Okay. We are all tired. Today we had 4 lectures, right? 2 more days of 4 lectures, then it's reduced to 3 lectures. It will be a little bit more time for all of us. Okay, see you guys in whenever, 10-15 minutes. Okay, but then you can relax. I'm not going to dump more knowledge on you, but I will answer your questions. Okay, and hopefully you will get all the presentations today. Okay. I can see how tired you are, right? It is when I go to a conference and I sit and I listen to people, I am so tired. When I'm working in my office for twice as long, I'm not tired. But when you're listening and listening and listening to listening, it is very tiring because you know you have to assimilate so much knowledge. वो बहुत ज़्यादा काम करा बहुत ज़्यादा हो जाता है हाँ बिल्कुल स्मेल जैसा है वो बिल्कुल सही बात है आई नो you know I always wonder because I work harder than you know in a normal working day. I'm never tired because I'm working at my own pace, right? और जब किसी का कहानी सुनते सुनते बिल्कुल थक जाते हैं और कॉन्फ्रेंस में क्या होता है हर एक घंटे में कहानी भी बदल जाती है and then you have to rearrange your brain to understand that new story, right? And it is very tiring. मुझे याद है when I come back from the conference I'm just dead for a day, like before I can read. And that happened to me as a student also. Like I would come back from a conference and I would just sit there कुछ नहीं कर रहे हैं trying to figure out आखिर मुझे हुआ क्या है ना आज बहुत ज़्यादा हो गया पर यू नो आई वाज टोल्ड कि संडे को आप लोग का क्लास कर दे मैंने कहा नहीं संडे को नहीं करेंगे आप लोग को ब्रेक चाहिए और मुझे उज्जैन जाना है दोनों का द रियल रीज़न you come to Indore and how could you not go to Ujjain? Yes. People come from all over the world to get to Indore, to go to Ujjain, and I'm in Indore. I have to go to Ujjain. आप लोग चाय पी कर आइए हम भी आते हैं आप लोग थोड़ा जोर से क्वेश्चन पूछिए जिससे सब लोग सुन सके, सब लोग बेनिफिट उठा सके. हाँ, रेडी? हाँ.
So you were told that for certain cancers, the metastasis will happen and they'll get a particular organ first. Like there's a tendency to get. So what governs that? Is there something?
I don't know that part, but I am assuming कि जैसे different kinds of cancers होते हैं ना, उनको different kind of जैसे हम लोग हैं, हम लोग को कुछ environment is better than the other environment. So breast cancer metastasizes preferably in the bone, for example. So bone environment is more conducive to their growth. It could be also a proximity issue. Liver cancer generally lungs में जल्दी जाता है. But it could also be that lung atmosphere is more conducive for that liver cancer. So like I was telling you initially when I started, I was an organic chemist, right? So I had no knowledge about biochemistry, glycolysis, nothing. So I used to naively think cancer is cancer, one cancer is same to the other cancer. That is the most naive thing to think that. So every cancer is different, every cancer responds differently, every cancer, so they like different kind of external signals. So that is why they tend to. But having said that, many cancers, they metastasize to the bone.
Another similar like attached doubt. If there is metastasis happens, so from the organ of origin, the cell will have a particular structure that is suited to that organ. So that cell will metastasize to some other location. So what would be the changes? Like if it goes from, let's say, breast cancer to bone cancer, to what will there be significant morphological change, or is it like a intermediate state where it works in both places and it's adaptable?
So one thing about cancer is that, remember when I was telling you TNM staging yesterday, right? Tumor nodes and this thing staging. So one thing they look in cancer is that how differentiated it is. So suppose hepatic cells look like this, right? Hypothetically. So cancer, when it starts, it will look like this, and then it will look like this, then it will look like this, and then it will become more round. So it will lose the shape of that thing. So this is the differentiated stage, right? So from differentiated, it becomes less differentiated. It has changed its morphology. Now they are more round on top of each other. So when the pathologists look at some cancer to tell how aggressive it is, if it is differentiated, it's good news because it is still looking like normal cell. The less differentiated it is, the more aggressive is the cancer. So by the time it metastasizes, it's no longer that original thing. It's completely different structure. So this will go and this will metastasize somewhere else, right? So that is one aspect. Because this cancer, this metastatic, these cells no longer look like the parental cell. Okay, and when they go to the bone, they don't have to look like bone cells or that because it's so metastatic. So that is one part, and that is universally true across the board. But having said that, remember I told you about EMT today, epithelial to mesenchymal transition. So one of the ways cells metastasize to other organs is that they have this epithelial morphology, right? And then they become completely mesenchymal like this, and they squeeze through hole. So this is called epithelial to mesenchymal transition, right? And they go somewhere else and lodge. Then there's a reverse process which answers the questions. Mesenchymal to epithelial transition. So they become more like that. Now they will differentiate. Then they will form more of those kind of cells, right? They will put their anchor down and grow a whole colony. Then some of them will become mesenchymal again, and then they will go to another thing. But they don't have any pressure to look like bone cells or anything because they are not trying to merge with that. They are so aggressive, they can be on their own.
One more small doubt. Does the division, like, does the differentiation within a tumor cell show some sort of, is it indicating towards the division of labor possibly, that they have separate functionalities and they all act together in some sense? Or is it just morphology randomly happening?
No, their morphology is different because, so when a cell is, so let me go back few steps here. Why do cells differentiate to start with, right? So when there is a zygote, right? So it starts with a single cell and then it divides and divides and divides. And then the cells, there is a division of labor, the cells differentiate and different organs are formed and everything. Cancer cells are not doing liver function, right? They don't do anything that the liver cells do. Their role is to just proliferate. They are not contributing to our physiology or anything, right? So they don't have to differentiate at all to do any function. What function they are doing in our body? Nothing. They are invaders, right?
I
They
was
are
talking in a sense that they are cooperatively working towards proliferating better, I guess. I think that was a linked metabolism in some sort. Like they are doing something like that.
cancer cells, when they help each other, that is the example I showed yesterday, right? The hypoxic cells are giving
Yes,
lactic acid.
Yes,
So if they are working together, they are only working among themselves to survive. They have nothing to do with the organ.
it's not a specific process they are doing, it's just transmission of substances they are doing. That's why they can need specific structures like
Yeah, and their abnormal structures allow them to escape from their abnormal structures allows them to squeeze through spaces to invade that. So they don't do anything for the good of us. They are just invaders. Yeah, they are just bad. There is nothing good about cancer. There's not even a single thing good about cancer. Our body is not getting benefited in any way. At least we don't know that in any way they they steal stuff. They will make these long tubes, steal stuff from normal cells. Yeah. And then you know about exosomes. They will release these small vesicles filled with, all the bad stuff, and they will go and invade neighboring cells, unsuspected cells, and then they will make them carcinogenic. So it is all they contaminate the neighboring cells also. So it's really all bad things. Yeah. And they are so powerful that they don't have to differentiate to hide themselves. But then they are also, it's all like they express these receptors, right? To quieten things. So they will have all these fake receptors to fool the immune system. So their whole energy goes in protecting themselves and proliferating. Yeah.
Ma'am, I have one question. Ma'am, we talked like in case of synthetic lethality when BRCA mutation is there. So we have one option of like using inhibitors. But ma'am, PARP enzyme has different isoforms, and there comes a question of isoform selectivity. So do we have a chance like delivering BRCA, like functional copies of BRCA in like thinking about gene therapy instead of using inhibition approach?
So the thing is that it is much easier so synthetic lethality is relatively a new concept, right? So if synthetic lethality works, you can have a very quick response like with PARP inhibitors, right? And a huge percentage of cancers are BRCA1, BRCA2 deficient. So when you develop a drug like olaparib, right, you can just immediately commercialize it, and worldwide it helps. There are so many tumors which have BRCA1, BRCA2 mutations, right? So that is very easy. You definitely can do gene therapy, but gene therapy is very complex process because you have to take out the cells from the same patient, then you have to engineer it, then you have to put it back in the patient, right? So it is so expensive. It is so expensive. And it may take very long time to do the whole engineering part and put it back. And then you have to also think, I don't know how many times you have to do it. Like probably one time adding the gene is not sufficient, right? So if you can make a drug which is orally available, or even if it is given intravenously, that is preferable because it is more cost effective. But gene therapy, people are working towards gene therapy, and someday it will become more and more and more accessible. as we learn about it. But gene therapy is definitely possible. So if you activate P53 or if you give back BRCA1, BRCA2, one day it will become very easy. People don't have to undergo all of these operations to remove the ovary or remove the breast. They can just reconstitute BRCA1, BRCA2, and it is going to happen one day. I am also very interested in muscular dystrophy because I know somebody who has it, right? In muscular dystrophy, you have to do gene therapy. So they there muscular dystrophy is a really devastating disease. The person cannot get up from the bed. It happens to younger people. So imagine your whole teen age, 20s, 30s, you just stay on bed because you don't have strength to get up, right? So that's very sad. Like the time when you're supposed to have maximum energy, you can't even lift your hand to do something. So, but relatively small percentage of people get muscular dystrophy. So companies are not interested in investing a lot of money in it because they don't make so much profit. So the therapy trials are still going on, and one day it will be realized, but the thing is, it is going to be super expensive. So how many people can afford that kind of thing? Like millions of dollars, right? So millions of dollars from your own pocket, how many people can afford that kind of therapy, right? And you have to have world-class facilities for, suppose somebody has to take out your own cells to engineer, right? But trust me, it is going to become a reality and it is going to become cost effective eventually, right? People are going to be able to afford it, and this will become true someday. So right now, even olaparib discovery that it works was a big help in this field. But you definitely can do it, and maybe 10 years from now or 20 years from now, it will become a routine process. See, you guys know that people used to have, like people used to die in 40s and 50s before, right? In my own lifetime, I have seen median age increasing. So if you can see that, so you can imagine that it is going to improve more. But we are also going to create more environmental pollutants, right? Like Venkatesh was saying, these things were not there. So we create, then we find a drug for that, our own creation, right? This is the drama, right, which will be there. Go ahead. Yes.
And there are several classes of like DNMTs and HDACs available. So like I assume in cancer and other diseases there will be certain HDAC or certain DNMTs that will be like higher in expression.
That's correct.
So my question is that like whatever is available in the market, like the inhibitors, HDAC inhibitors or DNMT inhibitors, they are a global inhibition, like global HDAC inhibition, global DNMT inhibition.
That would be bad also, right?
Yes, and also it is reported that in our gut microbiota, the microbiota produces like short-chain fatty acid, which also acts as a HDAC inhibitor.
Yeah, yeah.
So, like, why is that so? It is very difficult to target specific HDAC or specific DNMTs. So all those molecules reported, these are all global inhibitors, not specific. So why is that? It is because of structural similarity.
That is because of the structural similarity. So, I don't work on HDACs, so I don't know much about the pockets of HDACs. But what happens among family members, that they have very, very similar pockets. So, suppose you take any enzymatic class, right? Any enzymatic class where there is a super family of proteins. What is super family? You have many, many, many members of that. And if they all use the same substrate, their pockets are going to be very similar to each other. So when you make an inhibitor, there is a likelihood of cross reactivity. So, for example, I work on kinases, right? And all kinases, they bind ATP. So all their pockets look very similar. So there is a big joke among the scientists in the kinase field. So when Gleevec was developed by Novartis, so when a company works on a drug, So when you guys work on something and you publish a landmark paper, so how many people are involved? Like maximum 10 people from a lab, right? Maximum 10 people, right? So 10 people are there. Usually there are 4 or 5 authors and one is the lead author, and then you have your advisor, right? But when a company is developing a drug, there are hundreds of people which come. So there are many scientists, many people who work on molecular modeling, biologists, and this and that. So the first author is not the person who did the main work. Usually the first author is the person who is right at the very top. He or she has not done any experiment, but he is the one who is running the whole program. So these are the group of scientists at the lowest level, at this level, who have done all the actual experiments. Then their bosses, and then their bosses, and then their bosses, and then there is the absolute head of that. So they all come global, like so many people are there. So the person who was the head, who ran the whole GLIVAC program, right? His last name was Fabrio. I know him pretty well, right? He was that head of the whole program. So all those landmark papers of GLIVAC, they are Fabrio's paper. We don't know all other authors, but it is Fabrio's paper, right? So he famously said, there is no point in making kinase inhibitors. They are going to inhibit all kinases. Now he tells himself this as a joke, that that's what I thought. Because that time people thought you could never make a kinase specific inhibitor for one kinase, because they all bind ATP, the pockets are all similar to each other. But GLIVAC is specific for BCR-ABL, because now we have discovered that there are some kind of pockets inside which are specific to different kinases. If you make an inhibitor, you can do it. Second thing is that kinases have inactive state and active state. So when they're in active state, they bind ATP. When they're in active state, they don't bind ATP. So what is the unique thing about that? In the active state, they bind ATP. So all the kinases in the active side, they bind ATP. So when they're in the active state, their pockets are very similar to each other. But when they're in inactive states, their pockets are actually different from each other, right? So if you make an inhibitor which binds the inactive states like GLIVAC does, you can make a specific inhibitor. So now coming back to HDAC and DNA methylases, it is possible to make specific inhibitors, but keep in mind these are relatively new field. Epigenetic itself is a new field to start with. So we have made inhibitors, but we are going to make more and more specific inhibitors. And also with time, we will know more substrates of each of these DNA methylases and histone acetylases. So then we will know, okay, this histone acetylates these things. This one does this thing. So which one is better to target? Do you see that? Then things are not that simple. Everyone has favorite substrates, less favorite substrates. So how much we should inhibit versus how much we should inhibit this target. So you can do a lot of fine tuning also. So eventually we'll get to better specificity, selectivity. Yeah.
Can you just tell us how can we confirm these can be another complication?
So this is a very easy thing to do, surprisingly. Okay, this is relatively easy thing to do. So have you heard of AlphaFold, right? So you can do docking with AlphaFold also, and you can predict different kinds of structures and that, and then you can modify your compound. This is something I have seen for the last 20 years. People are doing that all the time. We have done in our lab also. And we can predict new pockets, and then we modify the compound accordingly, and it really works. It really works. So suppose you identify a pocket, and you see that that pocket is a linear pocket, like narrow pocket, right? It is not like this thing, but a narrow tunnel is there, right? and your compound is there. So if it is a narrow tunnel, you want to add a simple chain here, not like a isopropyl group which is branched, right? So you make a propyl group instead of a isopropyl group, and that will be a straight. And if you can access that pocket, it will increase the affinity of that compound, right? And then you have to test it experimentally by making both isopropyl and propyl and see which one works better. So prediction can tell you, and then you have to test it experimentally. If you only do prediction, it's not going to work. If you make a lot of compounds, that is very good, but computational modeling, after you make some compounds and after you get some data, then you can also do computer modeling to see whether using computer modeling is this telling you the same thing that you got experimentally. Then you know that your modeling is also correct. Computer modeling is developed based on experimental data, right? So the more experimental data you feed in, the more you know whether it is the accurate representation or not.
Just one more question. So I was just curious about the glue. So you told that this molecule inhibits the alien bond catalytic side. So
In the inactive form. I did not tell you that at that time, but Gleevec actually targets the inactive form of BCR-ABL.
it doesn't interact, it doesn't disrupt the interaction between BCR and alien.
Nothing. So BCR is very far apart from the catalytic domain. If you remember the picture, BCR is on one end and catalytic domain is there. But because of the BCR, the way catalytic domain is inhibited, it is no longer inhibited. So it is in a different open conformation, but it still is inactive to a little bit. So what happens is that ABL is shut down in normal circumstances. When BCR-ABL is there, it is inactive. But Gleevec binds in such a way that it traps it in the inactive conformation. So this is something actually we were working on. So I was at Novartis at that time when this whole story was going on. So they had just discovered that Gleevec was, a $1 billion drug for BCR-ABL at that time. But they could not figure out why, when you can inhibit BCR-ABL better in the cell versus when it is in the purified active form. So they even discussed with me, can you tell me why this is happening? So lot of people did lots and lots of experiments, and the end result was that this Gleevec inhibits BCR-ABL and locks it in the inactive form. So it can never become active. So that is why it is really active in the cells. Usually when you purify the protein, it is much easier to inhibit a purified protein than in the cell. But in Gleevec, it is the other way around. That time it was not even known that kinases have this inactive versus active. The kinases have allosteric pockets. Nothing was known at that time. So Gleevec was very good in revealing the mechanism of actions of kinases also. It is involved in a lot of signaling pathways. I have a friend who has spent like 10 years working on ABL. I don't know the specific signaling pathways, but it is involved in a lot of different cell pathways. It's a very good guy. See, the theme that is emerging, that the cells, if they're expressing something, these things are good for the cell. That's why they are expressing. Okay. But over time, if they become overactive, everything overactive or underactive is bad. Everything has to have the optimal activity. So if you work very hard, you also have to take a break. That is very important. Everything has to be in balance. We have to have time to regenerate. So anything too much good is also bad. Too much, CDK5 is activated, you have really good memory. But if you sustain that activation for too long, brain is going to degenerate, right? You smell good things, after a while you cannot smell anything because the system has to regenerate itself. So Abel does a lot of signaling pathways. I don't even know all the pathways of Abel. So don't think for a moment Abel is bad. If you remove C Abel, that's really bad because they all have function in the cells. But BCR Abel is not a regular protein. Some kind of really chromosomal translocation is occurring, which should not occur. And that gives rise to this really nasty protein. C Abel is a good guy. So, yeah. Yes.
Ma'am, if there is a family history of inheritable cancer, like genetic cancer, so can we perform the gene therapy of
You definitely
grandpa or
can.
grandpa to
Definitely.
induce zygotic for blastocyst disease? Like often, so when two couples are in a marriage, so
You
when they
know,
want to have a child to reduce the chances of him developing a cancer.
you know, you might have seen TV shows where they are genetically engineering the offspring. So the child is very smart. The child is not prone to diseases. The child is a superhuman, right? So these kind of concepts are not new, but there are a lot of ethical concerns about tampering with the embryo like that. Huh? Yeah, CRISPR also has a role on it. So anytime you touch the embryo at that stage and you start engineering it, imagine that most of the people are having normal children with normal intelligence. Think about a scenario, and some couple, they are very rich, so they play with the embryo and their own genes are not that great, but then they create a superhuman because they have the money to change the genes and everything. So, so if that is happening, that is going to, uh, then the parents will start complaining that kid cannot be in school with our own kids because this is an artificially created kid, right? And there will be tons of ethical concerns there. So when you touch something at the embryonic stage to create that kind of thing, you have to cross a lot of ethical things. Some years ago, human fetal neurons were very common. So when somebody accidentally has miscarriage, or even when people get abortion, the embryo, their neurons are extremely useful in discovery for neurodegenerative diseases, right? So my friend who is an Alzheimer's disease scientist, he had access to that because he works in medical school. So he did several experiments for us in human fetal neurons. But then the ethical concern things arose and Congress voted against it. You cannot use embryo for this thing. They are actual life. Even if they are 2 days old, it is actual life and you are killing embryos which cannot defend themselves and things like that. So all that research tanked. So basically, in principle, that can be done, okay? And eventually it will be done, I'm thinking. You can fix the problem, and it is definitely possible to fix the problems at that stage. But having said that, people are still trying to develop therapies, even a person is adult, that can be fixed. So these days what they do, that sometimes women, when they are relatively old and they want a child, so women who have their children when they are younger, their children are less likely to have diseases. But these days women in 40s are also giving birth to babies. But if the women are in their late 30s and approaching 40, or if they are in their early 40s, there is a very strong likelihood the children will be born to these mothers who are relatively old in terms of childbearing age. These children can have Down syndrome. Okay, so this is at least in US, that this is a insurance approved thing, that these pregnant women have to test their embryos for Down syndrome. And Down syndrome is extremely bad disease, mental disease. So you don't want to bring a child with Down syndrome, because more than parents, it is the child who has to live that life with that. So in that case, if it is detected early, you have to find out. I don't know what is the policy, whether you can get it aborted if it is at early stage, or whether you cannot get it aborted. That I don't know the policy right now, because this is a very sensitive issue. But in some cases, it is allowed to test the embryo for really bad diseases. But I can assure you, sometime in future, you should be able to correct it at the embryonic stage. And in relatively near future, we can do BRCA1, BRCA2 fixing in adults. So we don't have to undergo surgery to remove organs. We can fix it. Muscular dystrophy, I'm really hoping you can do genetic therapy. Cystic fibrosis, you can do genetic therapy. Go ahead. I mean, gene therapy, not genetic therapy.
So, ma'am, about BRCA1 and BRCA2, they are maternal, so they told maternally. So, can they be treated in the ovary, the egg cells itself? Like, can we?
You can. That's what I'm saying, that it can be treated, especially for in vitro fertilization. You can treat the things, but they are not done right now. When you are tampering at that stage, you have to get a lot of approval. So that is definitely doable.
Ma'am, cause egg is not zygote yet, so that might be less unethical or problematic.
The thing is that the people who make the policy, they are not scientists, right? And everybody has their own view of that. So yeah, it can be done. It definitely can be done. But keep in mind, CRISPR-Cas9 is a very new technique. It did not exist 10 years ago. So now that we have more and more and more powerful techniques, we should be able to do these things. And you know what? I mean, this is my personal view. If we can do it, and we know BRCA1, BRCA2 causes this kind of horrendous life, I would change it at the X stage if I could. Because, if you know something, you're not making a superhuman. You're not cheating in terms of intelligence. You are just giving a person a healthy life. And if you can, see, we all take medicines, right? And medicine is not bad if we can keep it. We all take supplements. It's not bad. If you can keep your body healthy, why not? Right? We are not cheating by keeping ourselves healthy. We are helping ourselves, and we are helping everyone around us by being healthy, right? And we are contributing to society. So if somebody, for no fault of theirs, has the tendency, or they have inherited wrong genes, why not correct it? And I'm sure it will be approved, and it will become a common procedure. But keep in mind, this science is going on regardless of everything. You can always take eggs from some other organism and check it out. So science is not stopping because of that. And I know personally some really some outstanding scientists, like really the 0.01% of this world, who are just truly, they do all these things despite hurdles. They try really crazy ideas, because these crazy ideas one day will give you crazy good results, You guys are so tired. Go ahead.
Once this is one hypothetical thing. Like in elephant, we see there is a higher abundance of P53 protein. So
Really?
yes, ma'am. So that's why they do not have any cancer.
Okay.
So why can't we replicate these things to humans by using a promoter enhancer kind of thing?
You know what? It is very funny, because I don't know. I did not have access to internet today. But I read this today, and I don't know how I read it today, because I did not have access to anything. So maybe I was reading my own lectures somewhere, because there is too much information in all these slides which I don't go through. But I read it this morning also that too much of a tumor suppressor is also bad because the cells will not even proliferate. So if you have too much p53, our cells will not even proliferate, and we need cell proliferation for survival, right? We share skin all the time, our stomach cells are proliferating all the time. So too much 53 is very bad for us. p53 is phosphorylated during cell division to switch it off. So you don't want too much 53. So keep in mind, nobody is perfect. You have to use everyone at the right time for the right amount of, like, right amount, right timing, then they need to disappear. So too much 53 is also bad because no cell division can ever occur if there's too much 53. So we have to achieve the right balance. So think about it in another way now. Our physiology is so complicated, so many things. How do we actually achieve the right balance? You and me, right? We don't even understand what is going on in our body. How do we achieve the right balance? We have to do 2 things, 3 things, maybe 4 things. Not having too much stress. We have to learn that. Stress will happen. We have to learn how we respond to it, right? We can have a happy attitude about it. Or we can, there are always ways. You can go running, you can do something you like, right? Everyone has a different way, and we have to use different strategies to deal with it. We absolutely have control over our diet. That is something. And then no one in the world is that busy that they cannot exercise, right? We always have time to eat. We have to find some time. When you come to work, instead of taking the bus, I mean, of course, not from Indore city, but for short periods, you can walk. In US, they will run in the morning, they will jog in the morning, but when they go to the supermarket, they have to park the car. They will go round and round and round and round to park closest to the shop so that they don't have to walk even this much distance. But in the morning, they are running, right? So you can park your car at a distance and you can walk. We always park our car in the first floor so that we don't have to take the stairs. You can park it on some other floor and you can take the stairs up and down. If your office is on the 4th floor, you can take the stairs to go up. I went to Japan, right? And I spent 3-4 weeks there. And in Japan, there is elevator in every building. Not even once any single person will take the elevator. Not even once. Every single time they will take the thing. I live in US. We are used to taking the elevator even to 2nd floor, right? So, and I was wearing high heels, and I never wear high heels. Never in my life. In Japan, I chose to wear high heels. I hated climbing the stairs. But when everyone is walking up and down, what would you do? When you go to subway, nobody takes the elevator. Everybody, every single person takes the stairs up and down. So my husband said, he's very active. So I had no choice but to go up and down with him all the time. 1 month later, I came and everybody said, you look so different. 1 month of exercise for a person who never did any exercise. It was such a life changer. That's when I realized how good it is for you, And it was very easy, something I could not dream of. I loved it at the end of my trip. It was so nice. Like, I would take stairs every single time, going up and down. And the place I was staying in, the university, right? Metropolitan University in Tokyo. So what they have done, and only Japanese people will do that. So they have made a university at a very high elevation. So there are like hundreds of steps you have to climb. If you have to climb hundreds of steps, and then they have acres of land where the whole campus is there. Imagine, no car can ever go because it's hundreds of steps, and they have not made a road. So once you enter the university, you have no choice but to walk miles and miles to the place you are staying. So even if you're coming at 2 a.m. in the morning, you have to still walk to go to your place of residence. If you have to come out, you have to walk that much thing. So you are forced to walk all the time. No wonder. Your body is so fit at the end of the whole thing. And Japanese people are very, very fit people. And when they make the line, there is not even a single person who is out of this thing. In our country, the line has no relevance because there are people everywhere. You have to see the line there, right? And nobody takes. You come to US, every single person will take the elevator. In the morning, they will run, they will jog, and then they will not climb the stairs. They have to take the elevator. I am included in that, right? So my point is there, it is so complicated. We can still do exercise without understanding the physiology. Body will take care of it. We eat healthy food, body will take care of the rest. So the least we can do after we hear that is to really take care of ourselves. And trust me, it is like, if you live a healthy life, it is true also that healthy people also get cancer, right? I don't know why they get cancer. It is free genetic disposition or something. But it still helps tremendously. Very, very, very few people will get cancer if they live a healthy life. That is definitely true. And, cutting down on alcohol, never getting exposed to smoke. If you live in Delhi, you are exposed to pollution. I don't know what is the, how you can protect yourself from that. But if you don't live in Delhi, at least you are protected from that kind of bad thing. And this is very serious, especially about Delhi, like the kind of pollution is in Delhi. It's very serious. I mean, somebody will figure it out someday, and but at least people are recognizing it is very serious. So
But I have noticed that leukemia is more common compared to other cancers in kids and teenagers. So is it true, and is there any specific reason for that?
I know for sure that leukemia is more common in kids. That I know. I don't know why they are more common in kids. That I honestly don't know. Because I have a friend, he works on leukemia, and he was telling me the same thing, that you see these younger people with leukemia. I personally know a lot of people who have developed leukemia as childhood, in their childhood. I don't know the reason for that. Yeah, I really don't know that disease, why. It could have a genetic component to it, and it could be something is wrong, so the bone marrow is affected. I have no idea. Actually, I asked him the same question, that why it is so common, and I don't remember he gave me any clear answer. I would have remembered if it was known clearly. But leukemia is a very strange disease. Like some people do not survive, and some people live their life while undergoing treatment. Yeah, in some cases it can be fixed also. And there are many kinds of leukemia also. I told you about a student who had leukemia in my lab, right? And she is doing extremely well. And she was diagnosed with leukemia when she was 2 years old. So, but then she underwent some therapy in Singapore. She is from Singapore, and it is gone forever, like kind of thing. Like she doesn't, initially she had to get some kind of therapy every 3-4 months, serious therapy. She had to be hospitalized. She was out of, like she could not come to work for 2-3 days while she was recovering. And then she got some treatment in Singapore, and now she does not have to do anything. So yeah, I don't know much about exactly what is the reason and what kind of therapy or things like that. There's obviously bone marrow transplant, but that does not give you complete remission. Yeah. No other questions? All right, guys, we finished today early, 4 o'clock. Not bad. So I'll see you guys tomorrow, 9:30. Our host and all other people, right? They're not here. So, anyone knows where is Venkatesh's office? No. You know. Oh, you know? Can you call Venkatesh or someone? Oh, I have his phone number. I can call him. I forgot. Yeah. Oh, I cannot call him because my WhatsApp phone is not connected to cell. Yeah, that is great, good enough. Yeah. Where is ladies' room here? Yeah. Do you guys know where is ladies' washroom? The washroom for there?